gotta be ready. All right. Hello, everybody. So before we move to a very exciting imaging session, organizers gave me the opportunity, for which I'd like to thank them very much. David, Karen, Joel, Sarah, and Vil. They gave me an opportunity to reintroduce to you our society's journal. And by this society, I mean International Society for Advancement of Cytometry. Can I move this thing so it... Oh, if I click more. Hide, okay. Oh, that was hide? No. Disable, hide. Oh, yeah. Okay, thank you. Perfect. So I would like to reintroduce our journal, the ISAC journal, Cytometry Part A, Journal of Quantitative Cell Science. And the reason for this reintroduction is that starting January this year, I became the co-editor-in-chief of Cytometry Part A. And sadly, after our conference in Edinburgh in May, I will be the editor-in-chief of Cytometry Part A after stepping down of Attila Tarnok, who could not be convinced in any way or form to give us another 18 years of his excellent service. He decided enough is enough. So taking over this role, I thought it would be a good idea to visit regional conferences like this one, which is one of the sort of leading examples of beautiful self-organization of cytometry community, to reintroduce the journal and introduce myself. So, as you know, I need to click. Okay, next. As you know, cytometry is linked to International Society for Advancement of Cytometry from the very beginning of its existence. And as you probably know, International Society for Advancement of Cytometry was not called that when it commenced. It was known as a Society for Analytical Cytology at the time. And cytometry started even before cytometry started, in a sense that our first editor-in-chief, Dr. Mael, was a guest editor for a special issue of Journal of Histochemistry and Cytochemistry, which reported on the scientific output from the third conference of what we know now as ISAC, which was held at Asilomar in California in 1973. And soon after that, or a few years after that, when it was pretty obvious that the cytometry community needs their own way of communicating scientific research, and being just a guest editor in another journal that publishes the conference reports is not enough. In 78, by the decision of the society, the new journal was created. And in July 1980, the first issue of Cytometry was published. And from the beginning, we were, well, not from the very beginning, but almost from the beginning, we were associated with Wiley, or with Alan Lee's company, which became part of Wiley later on. After Dr. Mael retired, then Jan Visser took over this cytometry. And then after that, it was Chuck Goldsby, and most recently, Attila Tarnok, since 2007, has been serving as the cytometry editor-in-chief. Cytometry was blessed with the absolutely amazing group of associate editors from the very beginning. I hope that you recognize some of those faces. I think if you don't recognize anybody on those pictures, you shouldn't be in this room. You should just leave in shame. You should recognize at least one person, but I'm absolutely sure that well, at least the FlowTex committee members recognize everybody. Obviously, we have Marilu Ingram. Some of you in this room are Marilu Ingram scholars, which is a distinction awarded by Isaac to dynamic young researchers with promising careers. We see Len Herdenberg, Zbyszek Darzynkewicz, Howard Shapiro, and Donna Arnjovin. And there were many, many others over the years. This is the masthead of the first issue of Cytometry from 1980. And these individuals were among the group of associate editors. So as you can see, we have tremendous tradition, which obviously will have to continue. And this is going to be a tough task because the publishing market is obviously incredibly competitive at the moment. But we will try. We'll try our best. So what are the 4 things that you need to know about cytometry that you probably already know? It is a peer-reviewed journal. It publishes original papers. It publishes reviews. It publishes technical notes, not only on the subjects which are related to flow cytometry. It actually has to be underlined and emphasized and screamed out of the rooftops that we are single-cell analysis journal. So whether it is flow cytometry or image cytometry or high-content screening or any single-cell omics, we welcome contributions which cover those areas. And this actually aligns with the tradition that we had from the very beginning. All of those individuals, they worked across multiple different single-cell analysis techniques. And cytometry from the very beginning was very welcoming to other fields that recognize this specific way how cytometrists think about biological reality, which is based on analysis of populations, analysis of quantitative statistical information about cells, rather than just reporting examples or average means. So we still focus on rare cell subpopulation, tracing cellular lineages, documenting complex cellular processes, and so on and so forth. We don't cover clinical field per se. There is a sister journal, which you are probably aware of, Cytometry Part B. But if there are new methods that may impact the clinical cytometry, but the research describes, I would say, fundamental technical aspect of this new methodology, this type of papers and reports are absolutely compatible with what we want to cover. As I mentioned, we accept original research, technical reports. We accept kind of specialized papers like reports from core labs describing specific practices that core labs follow. And this is another unique aspect of cytometry, which makes us different than many other journals. And last but not least, and I already mentioned this, we are the official journal of International Society for Advancement of Cytometry. And I'm, again, emphasizing this because even though we are published by Wiley, the total editorial and content control is with the society. So we are now, as you may know, facing the reality of huge changes in publishing industry. Many editorial boards of journals owned by big publishers like Nature Springer, Elsevier, or even Wiley, engage in what I would call a mutiny against the publisher because they were more and more controlled by the publisher. We are not one of those journals. We have a partnership with Wiley, but we control the journal and its content. So what is the plan for the future? Obviously, as I mentioned, there are lots of challenges. So we have kind of a strategic vision and strategic plan. I don't want to bore you with details, but the few important elements that I wanted to mention is starting with number one, elevating community interaction. we want this journal to be truly a journal of not only ISAC community, but all the cytometrists around the world. So they feel that the editorial staff, that the reviewers, associate editors really represent strongly the interests of the community and understand the field, understand the peculiar nature of the field and particular challenges that our field has. So I cannot promise you that it will be always the case. We're working on this, but you're more likely to engage in a constructive, I would say, dialogue with the reviewers when you submit your flow cytometry work or cytometry work in general, single cell analysis work. If you submit this work to cytometry than if you submit it elsewhere. It's very likely that the reviewers are going to be your colleagues with whom you have a beer during the ISAC meeting. And this creates a form of camaraderie and community, which I think we would like to expand. What we definitely want to work on is significantly reducing the time of processing the papers because it's unacceptably long at this moment. The average is around 50 days. We want to make all of that faster, but this requires your engagement, and I will come back to this point in a moment. We are adopting new approaches to presenting scientific research. Obviously, one of the most successful, relatively new format that we have is so-called OMIP, which is Optimized Multicolor Immunofluorescence Panel type paper, which is actually a very detailed method presentation paper. And we're working on some other papers which would be more visual and not traditional like the regular technical reports, allowing you to improve your practices in cytometry laboratories. We obviously want to expand global influence. Everybody does, but in our case, this is much more complex and complicated because there are certain cultural differences between the culture of publishing in different countries, and we want to take it under the consideration. And again, I don't want to bore you with details, but we are working hard on making sure that we become more global. And then finally, we want to integrate the journal more into the educational initiatives and outreach of ISAC. As you know, there is a CytoU. There are many other initiatives that either the ISAC leads or is led by the regional conferences like yours. And we want to be the sort of a voice of communication for those initiatives. So a couple last slides to conclude this short introduction. Why should you volunteer to review papers submitted to Cytometry? That's extremely important. All the journals now suffer from, well, a lack of reviewers, lack of engagement. People get invitations. Every other day, you probably get invitations to either submit a paper or review a paper. And the chances are that you reject all of them, right? Because you're busy, you don't have time. Well, but this is a paper, this is a journal, I wanted to say, that truly represents our community. So if you reject those invitations, you are, well, hurting the cytometry community, yes, and you're hurting yourself in the long run because you're not promoting the work that your colleagues are engaged in. Reviewing for cytometry also offers a unique opportunity for young researchers in terms of the personal growth, allowing them to basically follow what's new and hot in the field and kind of understand where the field is going. And of course, this is yet another avenue for volunteering, right? So just like you volunteer to co-organize, participate in a meeting like this, or present a talk, or in any other way contribute to your local cytometry community, you can do the same, engaging with cytometry on a more global scale, I would say. And even though this slide particularly mentions reviewing or volunteering to review manuscripts, there are actually two functions for which you could volunteer, right? You could either take a role of a reviewer, but if you are a more experienced scientist we would welcome volunteers who would like to take a role of associate editors as well. And in fact, some of you in this room, you may be approached by me about this role in near future. And so I'm warning you, you cannot say no. And obviously, the last thing is, why should you publish your results in cytometry? There are so many journals out there, and many of them have a much higher impact factor for the very simple reason that methods journals typically don't have a very high impact factor. They're less cited. And if they are cited, the dynamics of citation for the methods-based journals is much more spread in time, and impact factors measure just 2 years. So why would you give us a chance? Well, because of the type of audience we have. It's a very interdisciplinary audience. Cytometry is read by clinicians, is read by biologists, is read by engineers, and it's even read recently by bioinformaticians and people who do data science in relation to multiomics. So you will reach a very diverse audience. The journal is very easily accessible in a sense that because of the partnership with Wiley, you can find this journal pretty much in every academic library, right? So if you publish there, people will easily find it. They will easily find it whether your paper is going to be open access or not, because there is a very high likelihood that your library will have full subscription to Cytometry as a part of the package of subscriptions with Wiley. So even if you don't pay the processing fees to make your paper open access, it will be de facto open access by the fact that Wiley is one of the big 3 publishers, right? Next to Nature, Springer, and Elsevier. So this journal is very likely to be found anywhere. And the last reason is we are editorially controlled by Isaac. So the decisions regarding the publications are not based on the cold calculation, which is very often like, oh, what is the likelihood that this paper will be highly cited within the next 2 years? Let me think about it. No, decision is based on, is it cool application, great engineering, fantastic biology or not? It might be the more peculiar and weird example of the use of cytometry. But if it's scientifically solid, we will publish it. So that's another important point. So thank you very much for your attention. Please talk to me if you have any questions about cytometry. I will be here till the end of the meeting. With us, there's also Anna Belkina, who is Dr. Belkina is an associate editor of Cytometry, recently joining our team. So please talk to her too. And thank you very, very much. And I hope that you will enjoy the rest of the conference. Thank you.
Are you going to introduce him?
Well, don't fight over me.
All right, it's my pleasure to introduce Daniel. Daniel Vorcelle, the assistant professor in the Department of Pharmacology at Michigan State University. Joel and I first heard this talk at GLIFCA last September, and we both took one look at each other after the conference and said, we got to invite this guy to FlowTex. It was a very interesting talk. He does a lot of work in immunology, genetic engineering, all sorts of little things. And in 2024, he's named one of ISAC's emerging leaders. And so it's a real pleasure to have him. So let's please welcome him to FlowTex. Thank you.
Super exciting to be here today, mostly because it's 30 degrees back home. But really, I'm super excited to be here today, and thank you again to the FlowTex community for inviting me and having me here. So my talk, label-free cell sorting of distinct microbial populations based on autofluorescent and morphological phenotypes derived from imaging flow cytometry. Let's see. Laser pointer up. Okay. Or as I like to say, how do you measure population differences when you can't use fluorophores or afford an S8? So to start off, first we have to talk about virulence, which is the harm that a pathogen does to its host. So as you have an increase in virulence, you have a decrease in host fitness. And one of the ways that we can measure host fitness is by measuring reproduction or survival. And so this concept of virulence is really, why does it vary? Why is it that we have some strains of E. coli that are perfectly safe to humans and some are dangerous? Why are there mixed populations of pathogens that, some are more dangerous than others? And how does that virulence within that population sort of get maintained? And so the story today will kind of be the story of this project and how we first learned about it, the trials that we had, so that hopefully you can understand and try to apply for the same things to whatever systems that you might have. So this project started with the whale lab at Michigan State University, where they are looking at Daphnia, little water fleas, and their pathogen is Pasteuria ramosa, which is an endospore. And so these Daphnia here are about 1 to 5 millimeters in size, and the bacteria are roughly like 2 to 4 microns in diameter, so fairly small. And so first questions that I had was, okay, why are you working with Daphnia and Pasteuria ramosa? And as it was explained to me, is that number one, it's really easy to measure reproduction and to sort of, again, study virulence within this system because they reproduce every 3 days. And if you have decreased reproduction, they simply don't have eggs anymore. So it's a great little visual indicator of what's going on. Second is survival. They're invertebrates, so the morality of working with invertebrates. They have a very short lifespan. They're easy to maintain in really high numbers. Let's see. And last year is that the variation in virulence among the strains of Pasteuria ramosa. So there are some strains that kill quickly. There are some strains that permanently castrate the host. It's really, really complex bacteria that can do a whole lot of different things. But what's interesting, again, about doing this is that you can do population level studies. You can have a little beaker, and it's really cool if you go to their lab because it's just beakers stacked all the way up to the wall on every bench top. And it kind of reminds me of like sea monkey farms. But it's all these Daphnia swimming around, 500 Daphnia in a beaker. So now you can measure populational differences when you introduce a specific pathogen or a group of pathogens. And so really when you're considering the alternatives, they're not doing human trials, but trying to do the same things in mice and their costs and everything else. And the fact that once you infect these Daphnia that they turn orange, I like to think of it as that orange is the new black. So recap, again, as I'm learning about this project mentally, okay, they're studying bacteria within water fleas. So next question was, well, what do you want to do with these? And so first thing that they really want to start to measure is virulence as a function of absolute pathogen population size. So, right, what happens when is it that they are going to lose the eggs? When is it that you're going to kill things? Does this happen over time in a linear fashion? Does it happen in a logarithmic fashion? Is it a sigmoidal curve? Right, sort of the kinetics of virulence. The next thing is, okay, if we can have that with just one pathogen, what about within a mixed population? Is it that, all right, the blue pathogen within the group, the gold pathogen within the group don't really do anything, but it's actually just that pink pathogen. That's the one that produces a toxin that is related to, host survival, host fitness. And then thirdly, is it not just that simple? Is it even more complex? Is it that they're working together? Is it that once you have a high enough concentration of the gold pathogen that it interacts back with sort of this pink one, and then it produces the toxin? Is it that they work together? So these are sort of the underlining questions that the whale lab is trying to address with their system. Again, takeaway, all right, we're studying bacteria in water fleas and we're looking at kinetics. So next question was, well, it really can't be that simple. So what's sort of the catch here so far? And it's that these bacteria are morphologically diverse. And so it starts off with these dormant spores that are then eaten by the host. They get into the esophagus and attach in what we refer to as the mustache phase. And indeed, they go through these morphological changes where they look like little mustaches. From there, they actually sort of get castration. We see that the eggs disappear in the cauliflower stage. And these are actually the terms that are used to define these different morphologies, which I love. Followed by the maracas stage, the grape stage, and then we're all the way back to the host dies and releases a whole bunch more dormant spores sort of back into the population. And so again, as I'm refining what I'm thinking of with this system, okay, we're now studying weirdly shaped bacteria in water fleas and trying to look at their different kinetics. And so he said, so why are you coming to the flow core? What is it that we can do for you? What can we help you with? And he said, well, the goals of the system the goals of our system is to quantify the absolute abundance of the pathogens, to quantify the relative abundance, to isolate some of these distinct pathogen morphologies. But really the challenge that they've had so far is how in the world do they even start to do this? I'm thinking, well, go ahead, take the bacteria, you can streak them out on a plate, you can get the pure bacteria, you can grind up your Daphnia. We can see the differences in a cytometer. But, well, you actually can't grow these bacteria unless they are within the Daphnia. It's either spore that is dormant, it's chemically inert. If you're not familiar with endospores, they have found endospores that have been isolated in like salt crystals that are 25 million years old and they still grow. They're pretty darn interesting. But that's the catch. So we can't get you a pure population to start with in the study. Again, I'm thinking through this as you all are. It's like, well, great, we'll just use fluorophores, right? For cytometrists. Well, not quite. You can't use fluorophores either. There are no antibodies. There are no tags. Viability dyes do not work or penetrate in them. And even if they did, you're dealing with such a rare population compared to the Daphnia that is just sort of ground up. You're trying to find the bacteria that are within that and all the other bacteria that are in there. So that's what they came to us with. We've got a needle in a haystack that you need to find blindfolded. And if you can manage to do that, could you also isolate out some of these different morphologies for us? Now, as difficult as that sounds, we're all flow cytometrists, and that challenge alone just sounds enticing. And so we're like, absolutely, let's give it a shot. And looking at these different morphologies, my first thought was like, surely they're going to scatter light. Let's throw it on a cytometer. Let's see what it looks like. And really, let's just start digging in. And very quickly, we realized that these weren't necessarily project goals, but they were incredibly unreasonable project goals. Primarily because this is what it looked like as soon as we got it on cytometer. There are no clear populations, and this is on our 5-laser influx. And we started there for, let's throw it on. If we can see some distinct populations, we'll just try sorting them on a glass slide. We'll take a look. If that looks like it's in the right ballpark, then we'll kind of progress. And there were none. Anything here that looks like it's a clear little population, we isolated, we looked at it in all different fluorescent parameters. And no matter what we sorted, there was never any clear consensus on really kind of what we were getting at. And so with that, we told them to abandon the project, and that was it. Luckily, we had just we just won a grant, a USDA NEFA grant, to get an attuned Cypix. I know that we were one of the first ones in Michigan, and possibly the Midwest, to get one of these instruments. And we were really excited about this because it had 3 unique features for this project. Number 1, we had our bright field images. Number 2, the acoustic focusing. If you're going to try to find a needle in a haystack, it's great when you can go through that pile really quickly. And for the images, it can get 6,000 bright field images a second. So really, I like to think about this as not an imaging cytometer, but a cytometer that happens to have imaging. It's already there for what you're doing. You might as well use them. And lastly, the syringe-based fluidic system. So if they're trying to figure out the concentration of bacteria within a population and how that changes over time, they can get volumetric measurements. And sure enough, when we ran some samples, we were actually able to see these distinct morphologies. That was really encouraging. And one of the things that I was kind of blown away by was this is a 1 micron diameter little bacteria, and that's the kind of resolution that we're getting. Again, collecting things at 6,000 cells a second. We ended up some really, really huge data sets. For those of you that don't know what the software looks like, I just kind of wanted to walk through kind of how this process worked, again, how we ended up at the ability to sort out these populations just based on autofluorescence. So for every event that comes across, you get your corresponding bright field image. So my starting point was, great, I can see one of these little cauliflowers. Maybe I can go ahead then, in the software, load that up here. You can drop the resolution on your plots. And then when you go through and select all of those different little cauliflowers, I can say, show me where they are on the plots, which is really handy and useful, except when there's no clear consensus or trend on they're located. So kind of again had to go back to the drawing board and figuring out what do we do. The other great thing with this instrument is that it has automated image processing. Not only do you get the images right away, it'll draw the mask on there for the images and derive 26 different imaging parameters for you for every one of those images. So again, if you're like me a year ago and this was just all gobbledygook, or not having an immunology background, it sounds like people talking about CD markers. This is what the imaging parameters are. So first one is shape-based features, circularity, it's eccentricity, and these are a couple of examples from the training slides that come with the Attune, as well as intensity features. The one that we probably use the most is entropy. A little bit difficult to kind of wrap your head around, but it's sort of the checkeredness of the image. So in the top here with the red blood cell, it's monochromatic. So that has an entropy of 0. Whereas the cell on the bottom, we can see that there's a lot of little white and black splotches all over. So that would have a much higher entropy. And the reason for this is we said, okay, if I just wanted to try to identify the cauliflowers, the grapes, and the spores, what are some of these imaging parameters that I could use? Trying to think through this. And so first we're like, okay, well, entropy, right, could probably distinguish those cauliflowers from the spores. They certainly seem to have a lot more checkeredness. I could probably use circularity to go from the spores to the grapes. And again, entropy to go from sort of the grapes to the cauliflower. Surely there must be a set of imaging parameters that we could use to identify these specific morphologies. And so again, going right back into the software, it automatically processes the images for you, and then they just show up just like a new detector. So I can just go through then and say, all right, on the x-axis, let's go ahead and look at that entropy. Let's go to look at circularity. And when we did that, sure enough, there's a little small population up there that has the highest entropy and is really circular. I can draw a gate around it and then say, show me all the images that are right there. And sure enough, we found the cauliflower. So we knew that we were sort of on the right track. But we needed to find more morphologies. And so when we started to consider that we're looking at 42 fluorescent parameters, 6 light scatter parameters, 26 imaging parameters, we had to go to high dimensional reduction. The interesting thing here that I want to note, we're all used to using area. When you start getting into this with autofluorescence, width actually becomes really important. In fact, if you go to any of your current data, I bet if you went and gated height versus width on forward scatter for your doublet populations, you'd see that there are unique populations there already. What's cool is that we were able to see the morphological distinctions right away to these things that you might have always assumed were artifacts. So take another step back for high dimensional reduction, because I know that this is something that I was really intimidated by. These really cool, complex looking plots, but beyond them looking really cool, what are they? So when I like to explain this to our users, I like to think of it as how my 5-year-old son would kind of work with this. If I cut out a whole bunch of different shapes, put them down onto the table and ask them to sort them out, there's the way that we would do it as cytometrists, right? I could make a plot that is size, maybe number of sides. I could start grouping things together, width and length, height and base of the triangles, and I could eventually sort out these different populations. What high dimensional reduction is doing is exactly what my son does, and he's just going to group them on the table with the things that look like they go together. So you're taking something that previously required a lot of dimensions and a lot of plots, and you're just describing it in a 2D format. What's really cool about this is now if I say had an untreated sample, again, condensing 5 plots down to 1, and a treated sample, it's really easy now to just show two plots and say, what's the difference? Oh, in the treated sample, there's no more rectangles and there's a whole lot more triangles. That was our goal with the high dimensional reduction, and in its simplest form, sort of how it works. So next we had to use FCS Express. At the time of this presentation, it's still the only third-party software that allows you to do high dimensional reduction on the CyPix data and use the images. Because we're using images here, the first gating strategy that we have to use is the isProcessed gate. This simply means, is there an image associated with that event? It doesn't always get a picture for every event that comes across, so you want to make sure that I'm only looking at things with images. And the isProcessed is not only is there an image, but was it processed through the AI software? Do we have those morphological parameters? Second one is particle count. You really can't do a single gate on forward scatter height, area, or even on side scatter with these, just because the populations are so messy. But it's great that I can only look at images that only have a single particle in them. So that just sort of narrows the field. From there, I can do cluster analysis. And again, it also provides the images. So as I'm looking at my clusters, it will show me the exact images that appear within every cluster. And so when you compare the difference of only using those imaging parameters to generate your clusters, only using fluorescent parameters to generate your clusters, or using the combination of the two, that's what we did to sort of evaluate it. You get a ton of clusters using the imaging parameters, but as we can see, you end up with a lot of debris, not just sort of the spores themselves. The fluorescent parameters do produce some pretty distinct clusters, but it's very hard to separate out different morphologies. Based on fluorescence alone, it's hard to separate out the spores from sort of the very early stages of the cauliflower, to separate out the grapes from, even smaller grapes or some of the spores. I still love that I can give a presentation talking about cauliflower and grape-shaped bacteria. But when you tie the two together, and one of the hardest populations for us to separate out were these little tiny grape shapes, they're pretty decent up there. And so again, working together with just autofluorescence and imaging parameters, you're able to separate these populations out. So we went back to the initial project goals that they had for us, right? Quantifying this absolute abundance of the pathogens. We go back to what we were doing with our UMAPs, right? With this dimensional reduction. And instead of my treated and untreated, what if I color coded them, right? So I knew that the red ones were just my untreated samples, the blue were my treated samples, and I just merged the files together. More simple again, and I can see, oh, look, those rectangles only exist in the untreated sample. Well, we did the same thing where we would run uninfected animals, combine that with infected animals. Any clusters that only appeared in the uninfected, I knew were clusters related to the Daphne, just the ground up cells. And everything else was going to be related to the actual spores themselves. So from very early on, we were able to go, great, we can identify exactly how much spores by concentration release the bacteria among all their different morphotypes were in the samples. And so now Dr. Whale's lab can start measuring the effects of population size and virulence with different strains. Is one strain more active than another strain? How are the kinetics differing? So knocked one off the list. Second one is the quantifying the relative abundance of the pathogen stages. And sure enough, when we use both the combinations of the imaging parameters and the fluorescent parameters, we're able to get distinct clusters that do correlate to the different morphologies. And we had to have the images to do this, because it's the only way to validate indeed that cluster is the cauliflower, it is the grapes, it is the spores. And so again, doing this, being able to merge the files together, we can concatenate them across multiple time points, and now we could actually start to see that you would get more spores over time, then you wouldn't produce them anymore until the later stages. We could see what the rates are of each individual morphology. So again, accomplishing sort of their second goal here of quantifying the relative abundance over time. Again, instead of sort of the blue, pink, and gold different morphologies, right? It's the spores, the cauliflower, the grapes. At what combination of these different morphologies do we actually get that effect, that decrease in host fitness? And so that then brought us to our third problem, isolating distinct pathogen morphologies. So now that you can find these things, can you sort them? And We don't have an S8, we don't have an imaging sorter, but again, it really seemed like there were autofluorescent differences that we could sort of capitalize on. And again, looking at just how varied and mixed this sample is, can we find these discrete morphologies and separate them just based on autofluorescence on a traditional cell sorter? So how do we do that? How do we start with that? Starting with a cluster that again uses both the imaging parameters and the autofluorescent parameters. It would be great if we could use something like Hyperfinder. If you're familiar with that in FlowJo, you can just draw a circle around your cluster and it will tell you the gating strategy to use. Doesn't work for autofluorescence, so we developed a new algorithm called the Slow, Tedious, and Manual Finder, where we go through manually and try to figure this out. The nice part about that is I can share it with you here today, and instead of having to learn coding or anything, you'll see that it's doable, and it's doable across far more systems than just this bacteria. So first we identify the morphologically rich clusters, right? That grapes are down here, that I've got a really cauliflower rich cluster up here. Number 3 is the spores. Then what we tried to identify was sort of just forward and side scattering parameters that we could use to separate out the different clusters. In some of the previous slides, there are spots where it's hard to separate out the two. Sort of this interface right here, it's difficult to differentiate the grapes from the spores, regions where it's difficult to separate out the spores from the cauliflower. So first we wanted to pick out some pretty distinct regions. Then we again looked at that population, looked at our images to say, did we find a pure region? What else is contaminating in? When we found that there were little bits of debris or morphologies that we didn't want, we went back to the cluster. Again, we're able to look at which regions seem to be really rich in the spores. If you haven't done UMAP or the dimensional reduction, you can spend days just adjusting all the different parameters. It's difficult to find something that purely separates it, so you get good enough. And this is really just using the default parameters on UMAP, really gets you really nice and close with an FCS Express. Then we just looked at heat maps. So if within this cluster, this one had the debris, this one seemed to be really rich in endospores, I then generated density heat maps for each parameter. And so if we look at this closely then, I don't want this cluster, I want this region. So forward scatter again is going to be good for separating out that debris from the rest of the population. Violet again, yes, there is enough difference here, right, that low on the violet could probably separate out things from the rest of the debris. Something like the blue laser detector, and this is blue laser detector 1, which has a gradient that kind of goes up and down, wouldn't be ideal for separating out these two populations for each other. So you can kind of quickly go through and say, all right, here are at least a few candidate parameters that I could use for separation. You go through another gating validation, where you look at that then, forward scatter and violet 1, and you try to find the regions that, all right, if I'm more to the left of this region, that's where I get more debris. There's a region over here to the right where I'm just getting the pure spores. This is hard, again, I think, for most cytometrists because we are not ending up with a gating strategy that is all of the spores. We are missing a lot of spores in this population, but we can find regions that are specific morphology and purely are that morphology. And so then we take it to the sorter. Again, difficult when you're using the influx, which doesn't actually use area parameters. It's all height. And so again, we base everything on height on the attune. But even then, you can see that it looks very different if we're comparing forward and side scatter for the exact same sample on the attune or on the influx. But again, it's close enough. You can find these different regions that look like they're about the same. Same thing here for just sort of the violet and the forward scatter with the UV and the forward scatter. You can pick roughly the same regions, even though they don't look the same. And as my grandfather would say, the proof is in the pudding. We look at our post-sort samples, and we were amazed by the purity that we were able to get. And so we run all of our post-sort samples for validation back on the attune again. We get an image with everything. And just on the gating strategy here, this is 97.53%. Just mature spores, and I included the images of this other bits that are over there. And they are things that I see just like when I'm running sheath fluid, right? They're just little bits of salt that are still within the sheath fluid, or other little bits and fragments, sort of the Daphnia that were potentially stuck to the spores. But we didn't just do it for the spores. We were able to do it for the mustaches. We were able to do it for the cauliflower. We were able to do it for the grapes. And all these percentages here that are based on gating, we actually went back. We've done these sorts 43 times. And then for the post-sort analysis, we downsampled every sample to 500 events and then manually inspected all of the images to confirm whether or not that image was the target morphology, it was not the target morphology, or it was debris. And we made this table classifying the post-sort purity. We got better as time went on, mainly because we added extra rinse cycles in between analyzing samples on the attune. It was literally just our carryover at this point that was affecting things. But you can see that we were on average achieving like 99% post-sort purity or higher. And again, this is using morphological parameters, but actually taking this to just a basic cell sorter and being able to sort things out. Beyond the initial scope of the project, we were able to do this for small grapes. So even doing it based off of the small grape here or sort of those larger grapes, we were able to identify these intermediate morphologies that I have since termed the stingrays. We even found morphologies that had previously not been identified. That's really cool when you think about it. This is an entire field of research, but when they're looking at this, when they're coming up with this life cycle image, right? These are graduate students, undergrads, looking in a microscope for a crushed up sample. How many bacteria are they going to look at? 50, 100? And now within a few seconds, I can get 100,000 images. So now you can look at a huge population. We're able to start seeing things that people haven't seen before. That alone has just been incredible at being able to fill in the holes and the gaps. So in conclusion, Dr. Will came to us with this project and said, hey, kind of a curveball. Can you do something here? Because the entire field is kind of being held back at this point. We need to be able to isolate these morphologies. We can't transfect them because we can't isolate them. We can't get pure populations. Now they can. Now they can do, specific transfections. Once they're actually out of the spore state, they can start adding just cauliflower, in to look at the virulence. But overall in this talk today, I hope that what I've demonstrated to you is the impact of imaging and autofluorescence, because in the field of cytometry, this is something that we're going to have to be aware of kind of going forward because of how impactful it is. So with that, I'd like to acknowledge the laboratory of Dr. Whale, who is right here. She is truly one of those great researchers that you love to work with in a core, somebody that's passionate, that comes to you for questions and answers and is always ready to tackle a new challenge. The MSU Flow Cytometry Core Director, Dr. Matthew Bernard, who allows me to take on these kind of projects instead of just sitting all day on the influx. So I can't say how much I appreciate Matt's support through all of these random projects that I get to do. The USDA and the NEFA grant that allowed us to do these types of projects, that allowed us to bring cytometry to projects that had never considered it before. Michigan State University, the Office of Research and Innovation, Osteopathic Medicine, Human Medicine, Veterinary Medicine, Natural Science, and Engineering, who supplement the cost of the instruments and our salaries for the users so that we can truly get out there and help to push research forward. As well as the Duffy Lab and John Megan at the University of Michigan for those illustrations on the life cycle. And really the Thermo Fisher team over the year and a half that it took us to do this, and I said, none of those dyes work. That's because they were great at sending us all these dyes, and I go, nope, nope, nope. At hooking us up with all of these chemical specialists to go, all right, what is actually in the coating of an endospore? Can we try to think of chemicals that would work? We'd be able to stain things. Answer was still no, but they really went the extra mile in just helping us to, again, push things forward. And so that, I'd love to open it up to questions. I put my contact information here as well, if you want to get in touch with me. And the thing that I want to leave you with is, I talked about this with the endospores, but this is just the tip of the iceberg. We're doing the same thing now with zebrafish cells, that you can see different cell types with just morphological and autofluorescent parameters, and then go and sort them on a traditional cytometer. I love the S8. One day I hope to have an S8, but I want you all to know that the sky's the limit here on what you can do. You can do this with protoplasts, the number of unique populations that exist out there just on autofluorescence. And we don't have a spectral. The acetune is not spectral. So we really can do anything out there as cytometrists if given the opportunity. So with that, again, I'd love to open the floor to any questions that you guys might have. I know it's hard with the first talk after lunch because I was there yesterday.
Anything online? I think it's cool being able to blend two things and being able to solve the problem you did initially, and then that opens the doors that you finished with to look at other areas as well. I thought that was really, that was the gill hook for me.
Yeah.
Yeah, this is cool. I like
Well, if you think of questions later on, by
it.
all
Another
means.
one?
We really didn't. So from a biosafety stand,
Repeat the
Yeah.
question for the online.
so let's see if I can remember it, even though you just said it. Were we concerned about the biosafety aspects of working with these spores? You know, did it cause any cross contamination with samples later on? You know, or did we have any issues with them in general? So in particular with our sorter, our service engineers have always told me that we had the cleanest cytometers. We've never had an issue, and that's something that we were always cognizant on. So part of our sorting protocols is to regularly take, you know, pre-sort and post-sort samples to check for any types of bacterial contamination. We have labs that regularly do, you know, sequencing afterwards to tell them as well if you see or detect anything that's odd. I think that kind of goes back to sort of the basic protocols. We're all kind of switching over to things like Citrinox as well for like our cleaning regimens on the sorters. I'm by all means a traditionalist with using Contrad. I do think that you need a harsher detergent to truly remove a lot of the biofilms and things that break up. It's also nice that on the influx, it's really easy to do a hot switch on the sample lines in case there are already issues, but we regularly monitored it and we never saw any issues. But great question, because I know that that is a concern for a lot of things.
We have a question from online, which is, do you need an S8 or would a CCD camera on a sorter work just as well?
So this order was just done on an influx. And in all honesty, I think that you could get by with most samples with just a violet laser, which is where most autofluorescence is occurring. The really interesting takeaway that I had from going through this project was, again, not just focusing on your area parameters, but the width as well, right? If you can really drop down your width thresholds, right, you start to see sort of the localization like of the fluorescence, or it's spread within specific events. So that really dropped out a lot of populations. Yeah, so you don't need all the fancy technologies. You just need, I guess, the willpower and the willingness to try something new a lot of times. And again, it's the flow cytometry community. I wasn't doing this 4 years ago. The only reason I'm here is because of people like David, where I had a crazy idea and a question, and I threw it out to the community that really just wants to help other people do great work.
Trying new things is always fun.
Absolutely.
We've got another one online. Have you tried spectral and mixing with the CytPix data using all the fluorescent channels open? And I guess, could you try it also with other channels open too? But and just to look at how autofluorescent that the populations are.
So no, we haven't tried to do spectral unmixing because we don't have any reference controls that we'd be able to unmix it from. That being said, now that we do have pure populations that we can sort, we wanted to run and make reference library controls to try to start separating them out. We do have a 5-laser Cytec Aurora. The issue is that the autofluorescence changes so drastically. So that workflow that you saw, I had to do every single time for every sample to determine what the parameters are. And depending on the feedstock that was used for the day, the parameters would differ. It wasn't always the violet laser 1 and forward scatter that was the best for looking at spores that day. It would change. I do think that we could aggregate all the data together and say, here are some parameters that are probably going to be good enough over time. But they did change enough. It's something that I think that we want to look into to see if we could try to extract those out, but I'm not confident at this point that it would be able to do it.
Let me follow up with something just based on that. So did I hear you right that the different stages going from spore to cauliflower to whatever, the autofluorescence is different between those as well?
Correct. know I would love to get my hands on a lambda detector spectral to run some of these just to see like, you know, how deep can we go? But, you know, I know a lot of fluorophores out there don't necessarily have all the instruments, but it shouldn't stop you from trying to do something new and to seeing if it's possible. And again, the community out there is great to help you when you have these questions.
All right, thank you very much, Daniel. Whoops. Oh, sorry.
Within this population, that's a lot of the new morphologies that we've identified and then been able to separate out. It started with, we just do spores, and it was like, yes. And then it's, okay, what are the morphologies that you care about the most on this chart? And they said, all right, well, if we could get the mustaches, we could do some things. The cauliflowers would be interesting, and maybe the grapes. And then from there, it was, we had such large data sets that we identified all of these subpopulations that then it was, I wonder if I can separate that out of this entire mixed bag too. And I think right now we can do about 8 different morphologies. And so it's just with larger data sets and different time points, I'm sure that we'll continue to expand it from there. Yeah, thanks.
All right, as we're starting to get the next speaker queued up. All right. All right, it is my honor to introduce our next speaker. Dr. Katy Patras is an assistant professor at Baylor College of Medicine with appointments in molecular virology and microbiology at the Keck Center of Metagenomics and Microbiome Research. The goal of her research program is to study host pathogen microbiotic interactions in the urogenital tract with a particular focus in group B streptococcus and uropathogenic E. coli. Her group uses newly developed models to study why individuals with certain conditions such as pregnancy or diabetes are more susceptible to urogenital infections, to test novel therapies and to establish the functional role of all the urogenital microbiota, with ultimate applications to both disease pathogenesis. I'm just gonna like get tongue-tied in the afternoon. Pathogenesis and overall human health. That's really gonna help during my talk in a couple. Anyways, I'm also excited only because I've got to see the pictures originally. This Katy came through the core and was working with one of my guys, Claude, and I got to see so much go through and so much excitement over some of these images that I really wanted to invite her here to FlowText. It's really an honor. And so without further ado, Katy.
Thanks, Joel. Thank you to the organizers for putting this all together. It looks like a great conference. My title is Tamm-Horsfall protein augments, neutrophil NETosis, and UTI. The alternative title could have been how to use imaging flow cytometry to get your paper published. Just to give you a little bit of background on some of the key words of that first title I gave, I study urinary tract infections, or UTI. This is an exceptionally common bacterial infection, around 400 million UTI cases globally each year, a leading cause of deaths from bacterial infections as well. I know these graphs are a little bit hard to read. Pink is female, green is male. This is really just to point out that the bulk of the disease burden is borne by reproductive age women, as you can see by this chart down here. And it's also a concern for antibiotic resistance. So UTIs are the second common cause of antibiotic prescriptions, and we're starting to see emerging antibiotic resistance for all uropathogens to all primary antibiotics that were used. So we're interested in understanding more about host responses to urinary tract infections as a therapeutic development platform. Okay, so what does urine look like during UTI? Hopefully you guys can see that this is not normal. This is very cloudy. This would be called pyuria, which basically indicates that there is a massive influx of leukocytes or neutrophils into the urine, and this is the hallmark of UTI. What do neutrophils actually do in urine? We think that they're helpful. I would say that the field is a little split based on our models, but here's just some a mouse model data showing that neutrophils come quite rapidly within the first couple hours post-infection. If you immune deplete a bunch of different immune cell populations, neutrophils being on the right here, if you deplete them, there are elevated bacterial burdens, so that we think they're important in controlling infection. Lastly, last background slide, this Tamm-Horsfall protein that was in my title. Has anyone ever heard of Tamm-Horsfall protein before? I'm getting some enthusiastic shaking. I did not know this until I started this project. It's also called uromodulin. It is the most abundant protein in human urine. It's made actually in the nephrons of the kidneys and pumped into the urine, and then we excrete it. We make quite a bit of it each day, 50 to 100 milligrams. It's pretty big waste if there's not a function, so we're interested in understanding more about this function. It's a glycoprotein, so about 30% of its weight is glycans, and it's been used in clinical literature as a readout for kidney health. So if there's a decrease in THP in the serum, it indicates that the kidneys are not functioning appropriately, and if there's a decrease in urine THP levels, it actually suggests that kidney stones are present because the most common or abundant protein in kidney stones is actually THP, just because it is so sticky and it aggregates. Okay, so why am I interested in Tamm-Horsfall protein if I study UTI? Well, there's been some interesting global genetics studies that have come out that suggest that there might be a role for THP in UTI. And so there's two different variants of Tamm-Horsfall, the gene in the human population. One is considered the ancestral allele, which is a T in place of a C. One is a more recent allele. And basically, the people with T make more THP, and the people with the C make less. And what's interesting from this particular study is that there's associations between regions of the world that have an increased abundance of the T allele in the population tend to have increased abundance of UTI. So it's suggesting that these two factors might be associated. So my question as a scientist is, does THP directly impact urinary tract infections? And so when I first heard about Tamm-Horsfall protein, it was because we were gifted Tamm-Horsfall protein knockout mice. So I had to read up about what that protein is. I'm a microbiologist, so I infected them with uropathogenic E. coli, the most common cause of UTI. And you can see here, looking at urine burdens, bacterial burdens over the first week of infection, the THP knockout mice have elevated urinary bacterial levels, indicating that they are more susceptible. We also did flow cytometry and looked at the immune cell population differences between wild type and knockout mice. What stood out to us is increased abundance of neutrophils within the bladders of the infected knockout animals, both at days 3 and 7 post-infection. This was absent in the mock infected controls, so not something that's at baseline occurring in these THP knockout mice. If we neutrophil deplete these animals with a Ly6G antibody, we actually absolve the differences in bacterial burdens between the wild type and knockout mice, really suggesting that the primary driver of increased susceptibility of the Tamm-Horsfall protein knockout mice is related to neutrophils or their function in these animals. Okay, so what are all the different possible things that neutrophils can do when they encounter a bacterium? So this BioRender, man, I had PowerPoint in most of my training. Now we have BioRender, so you guys are using BioRender, quite lucky. So these are, just a readout for the various different functions that neutrophils have: activation, degranulation, phagocytosis, various forms of cell death. And so in my postdoc, I systematically went through and tested how Tamm-Horsfall protein might affect neutrophil function in all these different assays. And so here's the sum of several years worth of data. We found that THP directly interacts with neutrophils. It actually inhibits a lot of neutrophil function. It decreases ROS release, decreases recruitment of neutrophils, decreases killing of bacteria. Doesn't really affect apoptosis or neutrophil mediated cell death. And then there's this really interesting observation that THP seemed to enhance neutrophil NETosis, and it didn't really fit with this inhibitory activity in the other assays. So the parts that made sense, this inhibition activity, we actually found was that THP has these glycans, like I mentioned, and the terminal glycan structure is the sialic acid. And various immune cells have these sialic acid receptors on their surface that are used for recognizing self. So all of our cells are coated with sugar, typically terminal sialic acids. Our immune cells have these receptors to basically recognize self and not explode. And so what we found is that Tamm-Horsfall protein binds Siglec-9, which is present on neutrophils. There's a mouse homolog as well. And this is responsible for all this reduction in neutrophil activity, the chemotaxis, the ROS, the killing. We actually showed that it was very specific to the terminal sialic acids on THP. We blocked the binding site for sialic acids on the receptor. We didn't see this inhibition. If we clipped sialic acid off THP, we didn't see this inhibition. So it was a very clean, clear-cut story. THP inhibits neutrophil function through binding Siglec. So we published that part. But the part that we didn't include, because we didn't really know how it fit with the story, is this observation that we see increased NETosis. And so what that data actually looks like is we took peripheral human neutrophils from healthy donors, incubated them with urinary levels of THP, and then in this particular assay, not cytometry based at all, we're just actually measuring extracellular DNA that's released into the media as a readout for NETosis. And what you can see is if we look at stimulated cells, so PMA is a protein kinase C activator that increases NETosis. In the presence of THP, we see this enhanced production of NETs. Okay, so just one background slide on NETosis. It's a recently, fairly new, in terms of 20 years or so, described neutrophil death pathway. There's several different flavors of NETosis. So the classic would be, of course, the first described. It's also called the lytic stage. Basically, a bunch of different intracellular signaling cascades occur. It is ROS dependent. It results in the nucleus breaking down, decondensing, and the nuclear contents being spilled and released extracellularly. It's usually accompanied by degranulation and plasma membrane disruption. So that would be this classical pathway. Now, the field's kind of split on what we call everything else. So you'll see a couple different terms. I use the term non-classical because it's basically not the classical, but other terms like vital NETosis or ROS-dependent NETosis appear in the literature. And this is typically thought to be a faster pathway. Bacteria can stimulate this pathway. It may or may not be ROS dependent. And instead, in this case, the plasma membrane stays intact, at least for some period of time, and the extracellular DNA is released through vesicles. And it's thought that perhaps these neutrophil ghosts, if you will, that have lost their nuclear DNA still might have the ability to phagocytose or degranulate or cause some other activation. And so it might be a different pathway. Okay, so we wanted to see whether NETosis was occurring in the neutrophils in our animal model. And this is where we started using flow cytometry. So we took urine from mice 24 hours post-infection, identified our neutrophil populations by Ly6G and CD11B positive staining. And then the two different dyes that we further looked at were basically a live dead stain, so the immune-based viability dyes indicating whether or not the membrane was permeable. And then this non-cell permeable nucleic acid dye, Cytox orange. So everything up here has extracellular DNA. Everything down here does not. And then everything on the right-hand side is dead. Everything on the left-hand side at least appears to have an intact plasma membrane. And so we see these four different populations. For simplicity's sake, these two upper quadrants we're calling the non-classical and classical forms of NETosis. There might be some other dead cell populations in there as full disclaimer, but there's more evidence to come. So what do our wild type and THP knockout mice look like? Well, we actually saw the biggest difference was in infected mice in this non-classical NETosis cell population. So cells that are alive but apparently have extracellular DNA accessible. These were higher in our wild type mice and decreased in our Tamm-Horsfall protein knockout mice, fitting in with our in vitro data saying that THP stimulates NETosis. We also saw an increase in live neutrophils in our wild type mice as well. We wanted to take pictures. So this is not imaging flow cytometry. This is just normal immunofluorescence. Again, looking at the cells that were recovering from mice when our 1 day post-infection. We're using nucleic acid myeloperoxidase, which is a granule marker. This citrullonated H3, this histone marker that's classic for NETosis. We threw on THP antibodies as well. You can see that we see evidence of NETosis with these Hooks positive myeloperoxidase positive strands of DNA in the neutrophil population. We also saw that in knockout mice. It was harder to find, but was still present. Okay, so now we wanted to see, before we go too excited into mice, is this something that's conserved in humans? Is this phenomenon that we're seeing in our THP knockout mice translating into human neutrophils? And so here is where we were using isolated human neutrophils from peripheral blood, again, incubating them with Tamm-Horsfall protein levels that they would see if they were in urine, and then inducing NETosis with PMA. Our gating scheme is a little bit different. So here, since we have a pure population, we're looking at extracellular DNA, that Cytox orange dye, myeloperoxidase, which we didn't do cell permeabilization. So the only way that myeloperoxidase antibodies can recognize MPO is if it is extracellular. So again, double positive populations here are undergoing NETosis or indicative of NETosis. And then we also looked at Hooks staining and the live dead stain to kind of separate out two populations, showing perhaps a cell permeable or membrane permeable population from a membrane impermeable population. And when we looked at our different conditions here, we also saw that, again, only under NETosis stimulation, it's not that THP is doing this on its own, we see an enhanced total NETosis as well as both forms of NETosis, both non-classical and classical, again, indicating intact cell membranes and cell permeable cell membranes. Okay, so what's the mechanism for this difference is one of the big questions that we had. The literature suggests, again, that ROS, the reactive oxygen species, is important for NETosis. So we tested whether or not hydrogen peroxide in and of itself can cause an increase in NETosis in the presence of THP. Just using exogenous hydrogen peroxide, we saw that that indeed did occur, specifically in this cell membrane intact form. We also showed that it was ROS dependent. So if we used ROS inhibitor DPI in the presence of PMA, we no longer saw an effect of THP at all in enhancing NETosis. So this is the part where we know ROS is involved, but we really wanted to advance what is being impacted. And so I was committed at this point. I wanted to say, okay, what else can we do? Transcriptional responses don't make a whole lot of sense in neutrophils. They usually already have a lot of their mRNAs already made. So we decided that proteomics would be the way to go. And so I crossed my fingers and hoped that, $16,000 would get me some good data. Here I'm showing you that the biggest effect in our 4 different treatment groups, this is across 4 different donors, again, untreated, THP treated, PMA NETosis stimulated, and then the combination of NETosis stimulation and THP. PMA by far was the biggest driver of differences in proteomics data, which makes sense. It's a pretty potent cell activator. When we looked independently at pathways that were different in the presence of THP versus absence of THP, in the unstimulated cells, we really saw a lot of mitochondrial-related proteins that are involved in reducing reactive oxygen species, which again fits with this SIGLEC9 activation, so that made sense. In our main test group, we got a lot of non-significant pathways with a lot of noise. The main hits that we saw, and well, you can read this up there, was related to neutrophil granules. So basically the THP-treated, NETosis-stimulated cells had more granular proteins retained, which might indicate a little bit more of that non-classical NETosis form that tends to retain those granules. But it wasn't clear-cut. And so I was really at a loss. I don't think I wasted the proteomics data, but it was pretty negative. And this is when I was trying to wrap up some of the other flow cytometry stuff that we had going on in the lab, and was talking to Joel, and he walked in and was like, have you taken like pictures? We have an image stream. You might want to look at these populations. And so I was like, sure, we're already, I got to publish this somehow. So we did imaging flow cytometry. And so just a little bit of the basis for why this might be a good idea is because it's emerged that during NETosis, neutrophils undergo various morphological changes. They reach this critical determination step where basically beyond this NETosis will occur. The cell swells, rounds, and then the nuclear envelope is degraded, and then chromatin's released, and you get this really diffuse chromatin swelling, and then finally cell rupture. So it's pretty good evidence that this should maybe show us something. There was also a paper published in Cytometry, Part A, that was very helpful to us. That was not a pre-planned thing. It's just I was like, oh, that's where this paper is. That had looked at using imaging flow cytometry data to try to quantify NETosis. And so it was a really cool paper. It might have seemed like a good idea. So we jumped on board with this. And how it works is basically, using the bright field images as well as various fluorescent markers and their intensities or staining patterns to separate the cells into different populations. And so in our healthy neutrophil, we typically have, the bright field area, all the nuclear materials within the bright field area. So there's no what they call extracellular DNA area, which is basically these nucleic acid dyes extending beyond the cell margins. And then healthy cells tend to have, these polymorphonuclear type morphologies and have a low Hooke's area. If you have a decondensed cell, everything's still within the cell, but the Hooke's area is a little bit more diffuse and a little bit larger area. And then once you get NETosis occurring, you suddenly have the extracellular DNA extending beyond the bright field mask. And then also if it's a lot of DNA material, you'll have a pretty high Hooke's area as well. We get NETs fragments. This is, of course, something that can be a little tricky with flow cytometry, getting a lot of debris, but we take pictures of fragments. This would be an instance where there's really hardly a bright field mask at all, but there's still some DNA material there, and so a lower Hooke's area, but still this extracellular area described or calculated. And then just a couple of other populations that we weren't really interested in, but were something that was reported in this first assay, is cells that have a really decondensed nuclei, typical of apoptosis, and so a really small Hooke's area, no extracellular DNA. And then this dead diffused cell, so again, another cell that hasn't undergone this membrane permeabilization yet, so a no extracellular area and a really diffused nuclear pattern. So it sounded cool. Could we take somebody else's method and apply it to our data? We were successful, which is really cool. I actually suggested them as the reviewers for this paper. I don't know if they did or not, but I was like, look, it works. We did it too. We added a couple things. So we used Cytox orange instead of Cytox green. We used MPO instead of neutrophil elastase, a lot of similarities. But what we added was actually a live dead stain. And this is actually something that we had included even before we started using this masking method. And what was really cool is that we saw all these different populations. So our NETs here was type 3, again, the extracellular DNA that extends beyond the bright field mask. We saw some NETs fragments, so very little bright field mask, but still plenty of nucleic acid material. And the two different populations that they had called as dead, this type 6 and type 5, we also detected high positivity of our live dead stain. So that was pretty cool. Okay, so that's what you guys cared about. What I cared about was, did it show anything? And so when we looked at the different types, we actually combined the NETs and NETs fragments types, so this type 3 and type 4 here, because we saw in some donors, we got more fragments, and in other donors, we got more cells. And so we think the kinetics of how an individual undergoes NETosis is probably different. And of course, we were treating these all at the same length of time. So we saw that THP plus PMA enhanced the frequency of the NET populations, as well as this type 2 population, which is the still alive cell that has a swollen chromatin pattern, indicating that those cells are committed to NETosis. We didn't see any differences in any other populations. This was actually something when we were also including on this as a control, just sialic acid, which if it's just the THP sialic acids meeting this phenotype, we wanted to see if that was the case. And we didn't see any of the phenotypes mirrored in that group, except for the circularity score. I really don't know what this means, except for, we see that cells tend to round in the presence of THP plus PMA, but apparently if you add sialic acid, you might also be driving roundness as well. Claude actually pulled this out with his eyes when he was saying that. He was like, I think they're more round. So that was pretty cool. And then we also took a lot of really awesome pictures of nets. So these are just beautiful nets pictures. And just a little bit of a summary slide. If you capture any of the story that I told, I know a lot of this room is interested in the technology, but some of you are, trying to publish papers. And so here's just a recap of, how we kind of told the story and used this imaging flow cytometry to tell that. So in the absence of THP, we get PMA activation. We get this cell to shift more towards classical NETosis in an ROS-dependent manner. If we have THP signaling through signaling like 9, we do reduce ROS, and this tends to drive the cells towards this rounding and swelling and this almost delayed NETosis that tends to be this non-classical form. And so we're really excited. We think that THP might be potentially some activities there that might be helpful in protecting against urinary tract infection. And with that, I'd just like to thank my lab, various Baylor cores, of course, including Joel's core as well as the proteomics core. And then I have a bunch of glycan data that I didn't show as well from the UCSD core and my funding sources. And I'm happy to take any questions. Thank you. That's a great question. None of the in vitro data we just did with pure, oh, sorry, I need to repeat the question. Did we account for pH or osmolarity with THP or when we're adding THP in our samples? And the short answer is no. Our THP only treated group kind of served as a control that we weren't just screwing up the cells by adding THP. We're also not adding a crazy amount. It's actually really soluble in the water that we add. When you start increasing salt concentrations, like in the context of urine, THP actually forms a gel, which is actually how the original description of the the protein by Tamm-Horsfall was that it forms this this gel layer in urine. So it's the mucoid layer of urine. So high salt definitely tends to gel, but is very soluble in low saline or water.
So looking at like the images as well, did you look at the morphological information, whether you could classify the same things just on morphology information alone from the image stream?
Yeah, we used, I kind of didn't nicely break down the method as much. So we used this gating scheme and each of the populations were called and we just pulled images representative of those populations. So here was our cells separated from our debris based on the Hookston area of the bright field image. This here, we're looking at the extracellular DNA area, which is the cytox orange and Hooks staining beyond the bright field image. And then depending on how bright the Hooks area was or intensity of the Hooks, pulled our two different populations there. And then everything else that was gated came down here and we separated Hooks area by the intensity of the cytox orange dye to get our other 4 populations. And we didn't even gate at all by the live dead stain, but the populations it tracks with what they had shown, and that does that answer your question?
Yeah, if you have the data, let's take a look at it.
Yeah, that'd be great news, I guess. They weren't that expensive of fluorophores, fortunately. But yeah, it just certainly could be other ways. I guess in here Hooks was pretty important to measure the nucleic acid, but you're right, there could be further refinement of the methods as well.
There's a question online, and I think it's a good one. Do you only see neutrophil populations in the UTI urine samples, or what's the abundance of other immune cells seen in urine? Normal abundance, and then also UTI.
That's a great question. Yeah, neutrophils are the predominant ones, especially within the first 24 hours. But then you do start to get, but they're not the only. So there's also macrophage populations. We actually get a mast cell recruitment and activation in urinary tract infection as well. So there are other cell types. I didn't show any of the flow data where we compared the other populations where we looked at dendritic cells, macrophages, mast cells, T cells, and we didn't actually see any other populations that were different. So that's what led us to focus on neutrophils, but there's certainly other immune cells there.
Do you know also if there's any like systemic release of other immune cells and inflammatory factors?
We haven't tracked systemic immune populations. We looked at bladder, kidney, and urine because the bladder tissue itself might be different than what actually migrates into the lumen. But there certainly are, broad inflammatory cascades that are upregulated during UTI, IL-1 beta, IL-6, IL-8, or in mice, KC, MIP2 are all upregulated, especially during acute infection.
All right, if there are no more questions, then this, thank you so much.
Thank you, everyone.
Really appreciate it. Thank you. And we are now scheduled for a coffee break for the next half an hour. So if people online will join us back in a half an hour, and the rest of us, let's go and have a little coffee and talk with some vendors.
Talks after this don't seem really excited to go home.
sorting technologies. So Yui received her PhD in cell and molecular biology from the University of Texas at Austin, where she studied the cellular mechanisms of neuromuscular regeneration in response to muscle injury and in aging. From 2011 to 2019, she served as the director of all 3 core facilities, flow cytometry core, the confocal imaging core, and histology and tissue processing core at Dell Pediatric Research Institute at the University of Texas at Austin. She then moved to the University of Pennsylvania to study the function of cancer-associated fibroblasts in tumor initiation, progression, and metastasis. Currently, she is a faculty in the Department of Microbiology, Immunology, and Molecular Genetics at the University of Texas Health Science Center at San Antonio and the director of the flow cytometry shared resource. Her research interests focus on modifying tumor microenvironments to increase the efficacy of CAR T cell treatment in solid tumors. And Joel is the director of the flow cytometry at Baylor College of Medicine with expertise in flow cytometry and related instrumentation applications and techniques. He has over 25 years of flow cytometry experience and has been director of a core facility since 2006. He attended graduate school in immunology and cancer biology at the University of Minnesota, leading to cell therapy research or cell therapy model research. Joel worked on isolation and differentiation of antigen-presenting cells for potential cancer vaccines. He was one of the first to differentiate, expand, and isolate a novel second dendritic subcell type, the plasmacytoid dendritic cells, using human hemopoietic progenitor cells. He trained on multicolor flow cytometry and cell sorting at the University of Minnesota's Cancer Center flow cytometry facility for analyzing and isolating numerous cell types. His work on isolating rare and fragile cell populations from human and mouse and experience with hemopoietic progenitor cells and stem cells led to being recruited to Baylor College of Medicine to support an active STEM and cancer cell research community. He continues to support BCM community and provide services and consultations. Joel, with the help of others, founded FlowTex user group in 2007, bringing it to a popular annual cytometry conference. Finally, he continues to bring cutting-edge research technologies to the communities, including but not limited to high-parameter flow cytometry, mass imaging cytometry, and imaging cytometry. I don't know who's eligible.
Whoever put that together needs to learn how to self-edit. All right, that was me. Yes, my bios. It's hard hearing your own bios. So Yui and I are splitting our talk slightly. So what we're going to do is I'm going to go in and kind of give you guys an example of putting it into a core facility and kind of how we implement it, where Yui is going to go a little bit more into it. I got to keep the S8 for about 9 months so it's just, I just basically was fostering it, where Yui actually adopted hers. Well, hopefully I'll get one of it. All right. So I hope everyone can hear me. So I really got a unique opportunity, like I said, to foster the BD S8 Discover. First, a little bit about me. We already heard some, but we do have quite a bit of instruments at the core facility, and it's a wide ranging of instruments, and we do like to try novel technologies at every opportunity. And I'm really fortunate to have a great staff because they really work hard at testing these novel devices out. I first encountered the image-enabled cell sorter through an email from Dr. Peggy Goodell. She's currently the chair of our molecular biology group, and she sent me this email that says we've got to get one of these, and she sent a link to that. Of course, this was back then; it was still Twitter, to a tweet. And so I followed it to the research article, and we obviously got to see the results here. You get to see this wonderful. They started sorting on pictures, and so I was quite interested. And it was because it was a novel technology. I was interested because it had both imaging and spectral cell sorting. It kind of took it a little bit further than what the paper initially showed. It had a very unique capability set, and it's based on a familiar technology. And what was really lucky is that BD chose me as a center of excellence so that I got to foster it for 9 months. We got to help them test out their rollout. So we got to give them feedback on everything from their delivery to their install and everything else. And we met with them on a weekly basis to give them feedback. And we got the first S8 in the wild, as I like to point out. It did make us kind of a little star for a little while when I went to Cyto and subsequently Glyphco. But for us, this was a very unique opportunity. We got to actually try and a chance to be on the cutting edge with very limited buy-in. All we had to really do is our time. For 9 months, we got to have it. My staff and my users got to experience the advanced technology. And this also helped us generate preliminary in-house data for potential grants, which we've already submitted one. We plan on submitting another, hopefully be successful on one of them. And so that took us to actually evaluating the technology. So this is a multi-component system. This has both spectral, imaging, and sorting. And this is pretty complicated. We all know sorting technology. It's been around for a very, very long time. Spectral is a little bit more novel, and the imaging is even more so. So we wanted to know, can it spectral, can it image, and can it sort? And how do they work together? Well, first, does it spectral? This is an example. Well, as flow cytometrists, the first thing we do on anything is put beads. So we put some rainbow beads on it, and we can see here, this is our 6-peak rainbow beads. You can definitely tell they're 6 peaks. And you also can see the imaging channel. The peaks show up in the imaging channel, quite bright. Then we went a little bit further. We started to test it in other methodologies. In this particular case, what you're looking at is a comparison of CD4 titration that we did. We basically took CD4 on various markers, did a titration, but also ran those titrations on other instruments as well. In this particular case, we did the Discover S8, we did the Aurora, and we did our Symphony AS, or A5. And the Symphony A5, just for those, it's the first generation, so it is just PMT based with 31 PMT. And you can see that that overall, they all performed very similar. There's a couple cases here and there where they differ a little bit, but not much. And in our rank testing, which I don't show here, really what it comes down to is the Aurora having more detectors and being an analyzer did a little better in the rank testing, followed by the S8, followed by the Symphony, which actually makes sense considering that the S8 is a self-sorter and not an analyzer. We did have the fortune of looking at some of the data that was run on the instrument at BD just prior. They put together a 38-color test with using in-house antibodies, immunophenotyping. Here's just an example of the run-through. I don't want to get into the weeds on this. What I just want you to see is that this is really performing and showing the populations as you would expect on other cytometers. We then took our own in-house panel. We weren't we didn't go all the way to 38 colors, but we do have a 27-color immunophenotyping panel that we put together. It's geared a little bit more to the tumor microenvironment, but we took it and then we ran it quickly on our Symphony. We ran it on an Aurora, and we ran it on a Discover. And it really, and some of you have seen my talk yesterday, I used this slide as well. It really did show up, and obviously this wasn't perfect. We should have gone back and tweaked a little bit here and there, properly, of course. But here you can see that really we're getting, in this particular example, we're looking at subclassification of NK cells, B cells, and Tregs. And they all compare very well together. And this happened in almost everywhere where we were looking at. We really did get similar results to our spectral or high parameter instruments. So it's spectral. So now does it image? Well, here's kind of a smattering of images, all of which I'm going to kind of show you in just a heartbeat here. So we were impressed. We were able to see images. A little background on it, although Yui is going to go more into detail on the channels. Basically, there are morphometric channels that get calculated instantaneously back, and it's based on a masking feature. So when you set up the instrument, you actually set up a mask, and then all these morphometric features kind of go hand in hand, of which you can sort on. In this particular example, honestly, my most interest of all Katy referred to earlier, because whenever we've seen on the AMNIS, imaging cytometer, we were always interested in circularity. It seemed like circularity was always like one of these differing factors. So what we wanted to do is like, oh, we're having an imaging cytometer. And so that was like one of our first things we were interested in. We're also interested in size. Whenever people come to the core, they're saying, oh, well, are my cells bigger or smaller? And we're like, well, they're bigger on the forward scatter, but does that necessarily mean they're bigger? So there's a lot of questions we were really interested in as we went along here. So just a little background on the image. Now, this is not typical imaging. This isn't a CCD camera. This is basically 104 beamlets and using a kind of frequency change and timing, they put together an image. It's an assembled image. And it basically, on this particular, even though the paper had 6 channels of fluorescence, this has 3 channels of fluorescence, or is it 4 channels of fluorescence? But this one has 3. And so basically what you end up doing is creating a stack of 6 images. So if you were to look at this, basically for every event that you record the images, you're actually gathering 6 images, and that's 130 KB. Now, as an example, here, if you had 50,000 images, that are 50,000 events, you'd have 300,000 images at 6.5 GB, or gigabytes. So you can see that these files get up pretty large, considering that in a lot of our assays, when we'll sort or when we analyze, we'll do anything from 1 million to 10 million events. So, and the imaging itself is a little grainy, because it is a reconstructed image off of a PMT technology. But the fact is that you actually can see the images, and you can get subcellular resolution, especially on bigger cells. I have a quick little smattering of what some of the features are here. Eccentricity, which is one of the things we're always interested in. The max intensity, and that's within the, kind of mapping within the cell. But really, and I'm not going to go through everyone, as Yui is going to cover a lot of it. Our biggest thing also was this correlation, and this is the interchannel correlation. So you could see how related 2 fluorescent channels are, and that's if you're like doing nuclear localization, which I'm going to show you in a little bit. First, can it image? Another thing that we were lucky to do is we had one of my previous employees was recruited to Slingshot, and through that we actually got a unique little introduction to a product, and this is just what's called Scatterbridge. We were interested in it because it's kind of intent is to be a scatter reference. And so you can go and bring it to different cytometers. And as a core, we're always interested in references. But it gave us a unique opportunity to image. And so in this particular case, I'm showing an example of it on our image stream, which is these are multi-sized particles that are more similar to cells than opposed to beads. And they're also different densities, so that they'll scatter on the side at different. So it should create 9 populations, which it does. On our image stream, you get very lovely pictures, but that is the image stream, right? And we see our 9 populations, corresponding populations. And then as well as in our FACS-Discover. Our FACS-Discover did the same thing. And these are the corresponding 9 grid populations. Now, I'm not here to talk about Scatterbridge. This is just new data that we came up with. I will say that my staff scientist is going to be doing this, and it's poster 138 at Cyto. So if you happen to be in Edinburgh in the beginning of May, please stop by and we'll taste some whiskey and talk about various scatter particles. All right. As I alluded to, can it size? Well, we ran beads. We took some beads on there. We took 7.3, 10.3 and 16 micron beads, put them through there. We did some analysis on it from what our calculated bead area compared to the what was measured in light loss by the Discover, and it correlated really well. And you can kind of see the bead sizes as we go on. Now we tried it with cells. The cells, for some reason, now this is early in the software. The cells didn't correlate on what we were looking at as right. So if we look here, you're looking on the middle plot that I just added. the middle panel I just added. Those are CD3 positive CD4 cells, which are generally a little right around 7 microns. And you can tell by your eye that they do fall right around the 7 micron bead in size. They're definitely below 10 microns. We did find that the size scatter on it was the most correlative. But some of this I think just has to do with how you set it up in the beginning. And I know they're working on it. So does it size? Kind of. Hopefully getting better. Does it sort? Well, that's the important question. We are a self-sorting facility. And so the first thing we're going to do is we're going to start trying sorting. So first thing we did is we actually have some people who have NF-kappa B that's tagged with M cherry, and then they activate the cells, and then we sorted the activated cells. And we did a post-sort check to double check as you do. We sorted the cells, and then we did our post-sort. It worked out quite well. We decided, of course, we're cytometrists, so we like to do beads. So we went to what we consider a standard, which is our RMAX testing. This goes back to some issues with the ARIAs on the rectangle versus square flow cells that you could see issues. So we're like, oh, well, we have the beads on hand. Let's run the test. So we ran on multiple nozzles, multiple size beads. And our RMAX came out quite well. RMAX is just an equivalent of that you're actually sorting efficiently, that you're actually capturing the particles in the sort. And it worked out quite well. We could not get above the 20. We couldn't get the 30 micron bead to work. Of course, we couldn't do that on any of the other sorters very well either. So we did not include that data. We then had an opportunity to do a fun experiment on sorting Golgi localized GFP. And in this particular example, on the left here, you see actually untreated GFP tagged Golgi. And then on the right, you actually see cells that have been treated with Berfeldin A, which breaks down the Golgi and actually allows the GFP to then disperse throughout the cell. So this gave us a really interesting thing to analyze and sort upon, because we could use this matrix that's called diffusivity matrix. I can say that word. Anyways, you can see it right here, and you can actually see that that if you look the treated, they're more diffuse because the GFP is diffused throughout the cell. On the left, on the other hand, the treated is more punctate and it's not diffuse. And this plot right here can be sorted on. You basically draw a gate up, set it up. I'm not going to show all the post sorts and everything on there because I want to also talk about our GFP tagged NF-kappa B that I alluded to. So then we took this NF-kappa B and then we actually started sorting on unstimulated versus stimulated as well. So on the left, similarly, you have unstimulated, where you actually can see that there's a more that you can actually see the localization. And now what this is this is a comparison of GFP localization to a DNA marker. So on the left is unstimulated. And so you can actually see some of the images. The more you see in yellow, the more co-localized, and there's not a lot of yellow. Now, if you look over to the right to our stimulated, that therefore the NF-kappa B being co-localized into the nucleus, we see quite a bit of yellow. And once again, this correlation score here is a parameter that we can sort upon. We did get to try some various fun things throughout didn't get to do a lot with it after this, but this is from Leroy Verstig, who's one of our FlowTex committee members, as well as a former speaker and everything else. And he works in the School of Tropical Medicine under Dr. Hotez. And we were actually be able to take this, and these are cells that are been cultured with T. cruzi, and we got to see. And the whole goal here was to see if we could see and sort upon compartmentalization. Were these T. cruzi within the cell, on the outside of the cell, or some other variation? Was there one, or was there multiple? And we were able to break it down. We didn't get to do a lot of work, but when it comes down to it, we were able to break it down by the delta center of mass versus the CFSC label on the T. cruzi. And we could actually, by this plot, although it doesn't give you a lot of data, break it down to whether they're internalized, whether they're centrally located in the cell, or they're external to the cell. One of my favorite ones we did was on circulating tumor rafts. So this is a xenograph model of human breast cancer. And what they're doing is they're looking for the circulating tumor cells after the implantation. So, and they're not only interested in that, but they're also interested in the myeloid associated cells with it. And what we wanted to do was to, one, analyze to see what kind of rafts there were and how they were associated with the tumor cells, and then sort the ones of particular interest. Now, in this particular case, we obviously could very easily sort out the cells that were positive for the neutrophil positive, GFP positive, and all that. I unfortunately I don't have the data here, but we were able to kind of correlate the clumpiness on this. And in some terminology, we call it swarming, where you'll get particles. You'll have more particles either closer together, farther apart, or closer together, or just random. And in this particular case, you can actually see on the left, we actually a little more clumping. And it really, the idea here wasn't to see the swarming and the clumping. For them, they were actually wanting to be able to sort these myeloid cells for potential downstream analysis, or to evaluate the technology to whether they were going to do immunophenotyping and see about variation. And this is still an ongoing project, at least in imaging. Hopefully we'll have an essay at some point to go a little further or to hear back from their DNA analysis from the sorting. So I've kind of gone over a couple different components here. We talked about spectral. Can it spectral? I say yes, it does. It does have 78 channels for spectral. So it actually has quite a few. Imaging, it does imaging. Is this going to be an amnis? No. We recently are testing the CytPix. Does it have that resolution? Not currently. Does it image? Yes. We can get correlation. We can do a lot of other things on it. We are hoping that the software comes a little bit more mature, so that we can then do a little bit more analysis of the imaging profiles. It's still for us a work in progress. Can it sort? We thought so. We actually tried it on a couple other things. I'm not going to give the data because it's not necessarily mine to give, but we were involved in an ABRF study which hopefully the data will come out, and this is where they're mapping all types of sorters. And it was a deposition assay, and I'll leak a little bit and say the S8 actually did a lot better than our ARIAs. So we started to question our ARIAs, which is, anyways, future directions. First of all, we still want to continue troubleshooting the cell sizing. We do have the data. They allowed us to keep the computer so we can actually do some of the analysis. Then we're also going into cell profiler and some other methodologies. We are going to kind of continue this and develop an image analysis pipeline via cell profiler. This is something we intended to do anyways. And like I said, we also put in a grant. We actually didn't get to play with the autofluorescence extraction as much as we wanted, although we still have the data. So we're hoping to get some time to go back. Also, I would have loved to do some doublet sorting, some immune synapse stuff. I mean, it would have been really cool. Unfortunately, it just didn't work out timing wise. So what this leads us to is that we did put in a CPRIT. This wonderful opportunity actually allowed us to put in for a CPRIT Core Facilities Support Award for a renewal on it and use this technology as the keystone. And as well as hopefully a shared instrumentation grant as well, we're putting up in the upcoming submission. Hopefully we will get one. I do have a lot of people to thank though, real quick. My team doesn't get thanked enough, I don't think. Claude, who did a ton of work on here. Obviously all my team contributed a ton. Madvi, Courtney, Mason, Paul, anytime you see a slide that has statistics, that's him. Amanda is my lab manager, Wego, and our newest member, Huntama. And of course, I mentioned Padmini. She is no longer with us, but she is our slingshot rep, and so we get to talk to her still. I will say that here's a picture of us on the install. We had a great group of engineers. Our local engineers are actually quite awesome. and I got to meet a lot of BD people and get a lot of attention I probably didn't deserve. And then here is one of our, I don't have the most up-to-date picture, but we do a Thanksmas for our core every year, which is like that in-between area after Thanksgiving. And we have a great core day where we come together as a group and have a potluck. So that's them. And that is it for me. And I think are we waiting till after you to take questions? Well, then I'll open it up for some quick questions. Yes, Anna. Yeah. That's on the oval. So if you notice on the image stream aspect of it, they're round. So here's the one, because the way that the data image is created, we are noticing some of these artifacts. Some of the other artifacts you'll know is kind of like these little ghosts that we have. We're really interested. This is one of the things we're thinking about on how we would clean up the images. And unfortunately, we are not, we don't specialize in imaging. So we are going out. We put in together for a no label little micro grant from our cancer center, and Claude's going to be working on some cell profiler data to kind of look at this type of thing, both for the image stream and the Discover and potentially the CytPix as well. I think this is just a lot of this is just artifact of the technology, and it's probably taken care of with software. I mean, yeah. The recovery, like, that was really good. If you saw on the RMAX, which is just beads, mind you, right? The RMAX study is really what it says on your efficiency of recovery, and it was spot on. We really did well. And matter of fact, on our deposition testing, and I apologize a little bit for the tip of the hat to our ABRF people for that group, but it worked out really well. Like I said, it functioned the best of all our sorters on the ABRF. This was a deposition testing on the slide, and then subsequent counting of beads, different size beads, mind you. So yeah, it was a solid sorter. I mean, when it comes down to it, it's slightly reduced in speeds. So your maximum on your 85 micron nozzle is 16,000 events per second. So you're not going to tear through it. It is, I mean, I always kind of hold as a basis of comparison the MoFlow, as I've pretty much think the MoFlow is probably the most efficient high-speed sorter, although sadly it's no longer made. Actually, I might not know that. Coulter may have brought it back. I thought they retired it. And then also the Influx then got retired as well recently. They're no longer selling them, I'm understanding, which is sad. I really was hoping to get one. So as of right now, my basis of comparison is the Arias. It does not go as fast, but it definitely is stable. The fluidics are more stable than the redesigned fluidics in the Aria. All right. Let me see if I can get out of this. I just close that and I'll get into it. Which one are you? This one? Nope. Why am I doing this?
In the chair.
Yeah, get out of it first. Then, we're not even showing.
Thank you, Joe, for a nice introduction into the world of imaging plus cell sorting. So, like he just mentioned, I'm gonna give you a little bit more detail into the imaging features on S8, because I think that's really what made it unique in the market right now. So, and for our research. And so I like, it's not moving. Click. Okay. And do I just keep on clicking? Okay, thank you. So for people who've been who have been doing sorting for a long time, in the back of our minds, right, we always wonder what have we been collecting, right? Especially, I think for me, and because I've been using microscope on the side to check my samples. So, so I think for a long time I've been whenever I meet with my reps and say, I thought, can we look at these cells while we sort? When is that technology going to be available? Right? And so 2 years ago, and we're very excited to see this paper published in Science and reporting that the technology is almost mature at the time, right? And it's a very beautiful paper. And then in the paper, they showed us they can not only imaging those cells, they can also use those imaging features to do cell sorting at a high speed. And they also, in the paper, they also show that the resolution of those images is high enough to be able to separate those, the different mitotic stages in cell cycle. And they also, of course, show that the nuclear factors and NF-kappa B transcription factors in the nuclei can be identified in the nucleus by staining nucleus. So here's the schematic of that system. So I'm just to give you a quick overview. How the imaging that's down in here is very different from the traditional imaging that's acquired with microscope objectives, right? So here in the center, as you can see, that's should connect it to the pointer. Yeah. So that's the traditional flow system. You have the flow cell, and the images are all down on one laser. It's on 488. So this 488 laser, first they split the beam 50-50, and it goes through a acousto-optic deflector. And to generate an array of the beamlets, those small beamlets, that's what it generates images by creating the radio frequencies. And the other half that's been actually split into a frequency matched beamlet, and then go through the acousto-optic deflectors and combine them, the two paths together, go through a 10x objective to focus the light before they hit the cells coming down. So at the interrogation point, we know, right? And the lights are actually collected. There are 3 bright field images that's generated from the scatter, forward scatter, side scatter, and light loss. And for this prototype, they actually have 4 fluorescence channels, and the GFP and the sort of around a P channel, and then a little bit redder on 700 APC or P sci-fi, and then the P sci-fi. So then the signals, of course, are either collected by PMT or the photodiode. And the images generated, or the features of the images, are actually incorporated into software and being utilized to make sorting decisions. So our facility is very fortunate to actually acquire the first S8 in Texas. And of course, many thanks to everybody at BD who twisted arm for us and made it happen for us. And so it's really exciting. And that's the system in-house installed in August last year. And our VPR office was super excited as well about this system. So they actually did a PR advertisement for us to the school, in the school. So it's really exciting. And so just a quick overview, as Joe just give you a very nice description of the system in much detail in the sorting, imaging, and the spectral side. So the system we got, the commercialized system, is the Phi laser. Right? It's a spectral sorter. And this, on the spectral aspect, it has 78 detectors. And I combine the real-time imaging, as I just mentioned, and it can sort up to 6 ways, just like the S6. And it can sort into tubes and plates. So, of course, I just said I'm gonna focus on the imaging aspects of the system because that's really what made it unique. So on the imaging side, there are 6 channels, 3 bright field images, 3 fluorescence images detectors. So for the 3 bright field, there's, I just mentioned, forward scatter, side scatter, and light loss. And then I'm gonna show you each image how it looks like actually in the system. And the 3 fluorescence channels, they're the peak is 534. The first one is 534. So of course that's the GFP 50 Alexa 488 range, and then there's the 600. So this one is PE because because you see the excitation has to be 488, right? And in the third channel is 788. It's the far red, and of course that would be equivalent to like PE size 7 fluorophore. And so I'm gonna go through each channel and how each images looks like in the system, right? So the light loss, the light loss is actually occur in the direction of perpendicular to the cell movement, right? So the cells absorb some of the light and generate the image, and it's so it almost looks like the bright field with the microscope. As you can see, this is the reconstructed image of each cell, and they have a very nice round shape. Right? And some of them have a dark ring that's in focus. So we're pretty happy with these images. And then side scatter, as we know, the side scatter is collected almost at a 90-degree to the laser entry direction. So, and the side scatter, so it's a reflection of the particles. So these the cells reflect. It correlates to the optic density and the granularity of the cell, and it also tells you actually the size of the cell from the imaging, as you can tell, right? So the next one is forward scatter. So the forward scatter, so these light are collected at the laser illumination path. So a small angle, so 0 to 15 degrees, right? And so these images, as you can tell, again, it tells you the size of the particles or cells. And it also tells you can discriminate morphological differences. And this is a doublet here, right? So, so we have been thinking about, so now we can already tell the difference from just by looking at bright field images. What can we do with them? And so there are 2 imaging features that I would like to introduce you before I show you the real experiment. And so the first one, as Joe just mentioned briefly, it's eccentricity. So it measures the elongation of those cells, whether they're elongated or they're round. So if it measures the shortest axis versus the longest axis. So if they're round, as you can tell, right, so the eccentricity is almost as 0. or if they're elongated, then they're almost at 1. Okay, so that's one. And the other imaging feature that we are going to utilize is called radial moment. So radial moment is a measurement of the spread of the signal. If the signals are are aggregates like foci, so the radial moment is on the small side. And if they're evenly distributed in the cell, then the radial moment is bigger. Okay. So first, we found out that the side scatter images are really useful for us to help us to separate doublets. So here's an example, like as we all very familiar with, and so we use side scatter area height or width, or full scatter area height or width to separate singlets from the doublets. But this is as good as we can get, right, with any traditional cytometers. And so with this system, if we gated out those singlets as we think those are singlets, and use the eccentricity versus radial moment, and we see that most of them are they are organized as just a flat line, right, in the plot, but there's some of them sort of curved up. So what are they? Now, with the images, what it tells us is that in this area, so most of them are singlets. Okay? And if we put our pointers around this curved up area, most of the events that we saw, those are doublets, those are weird shaped cells, or all kinds of stuff that's in there. So we can do as best as we can, right, to separate this, to gate out singlets, but with the imaging feature that can really give us more accurate the separation of singlets from doublets. So and the next example I'm going to show you that's really exciting is that, so one of our users, actually, they came up to me, they asked me, now you have this imaging sorter, can we do label-free sorting? And so they study endometriosis. So what they want to do is that because endometriosis, right, it's a worldwide problem, a major problem, especially in women, and in women, and so it takes a long time to diagnose, and most of them are misdiagnosed so far, and it's very costly for patients. So what they are proposing to do is that they will get these uterine biopsies from patients, and they want to separate the cells, and most of the cells in the uterine biopsy, they are either epithelial cells or the stromal cells, right? So for a long time, what they have been doing is that they have to label these cells first, right? Use Abcam to label epithelial cells, or Vimentin to label stromal cells. And so that takes a long time, right? And afterwards, they would isolate these cells, actually manually pick them, and move forward with sequencing. So it's very labor intense, and they want to shorten that period of time and also get more accurate results. So this is what we did. So we were very happy with the results. So just by doing no label at all, right, and they isolate the cells, put it into single cell suspension, and we use the eccentricity and radial moment to identify the cells. Let's look at the cell size, right? And in the higher radial moment range, that's the bigger cells. Those are epithelial cells. Okay. And lower range radial moment, these are stromal cells, much smaller. Very easy to identify them. And of course here, I have to show it because these are the part that we will discard absolutely. Yeah? So they actually took those cells back. They did RNA-seq with them and pretty much on that. Very nice. Yeah. So, okay. So that's just the bright field images. Now let's take a look at the fluorescence. As I just mentioned, the fluorescence images. So this is just keeping in mind all those fluorescence channels. They have to be excited on 488 laser. And so we have the green, that's in GFP channel, and then the PE, and then the PE size 7 equivalent acquisition emission channel. So this is an example that we did actually in the GFP. Actually, we used the FITSI and then the PE to label 2 transcription factors in the nucleus, in the cell, and then this is the drag file label to label nucleus. And the overlay, as you can see, we can again identify which cells actually express either both of the transcription factors or just one. And they color very nicely. So now if we combine all 6 channels together in light loss, forward scatter, side scatter, and then the GFP, the P, and the drag file. So we can overlay all of them and identify whether these labels are actually in the nucleus or in the cytosol. And so the resolution, as Joe just mentioned, right, it's definitely not there, not like image stream, but it's good enough for still for us to tell that to identify the location of your protein. Okay, so again, examples, and so that's really why we think that essay is so good. Sorry, what happened? Excuse me, sorry, I'm a little bit under the weather. So so anyway, so here's the first example that you utilizing the fluorescence imaging aspects of the on the assay. So this is just a simple sort on GFP, right? We've done that. Most of us done that. I've done that. So that's just the GFP tagged plasmid. We want to transduce to the cells, transfect the cells, and then we want to sort out GFP positive cells. That's it, right? So this and this lab, we sorted for them, and they wanted the brightest. They just want the brightest GFP cells. Fine, let's do it. Right. So this is ARIA always, right? And on ARIA, we did it for them, and top 5 GFP positive, and then the postdoc came back to me and she told me like, oh, I checked I used the Western blot to check my protein. It's not there. So what happened during the sorting? Huh? You want the GFP positive. This is the GFP positive cells, right? And so then I was like, well, maybe something funny happened during your passage. We don't know, right? And because she showed me her Western blot before sorting, after sorting. So let's take a look at these cells. But where are her plasmids? Right? So we did exactly the same thing, right? Just by looking at GFP, the top 5% with the imaging. So this is an overlay with GFP on the light loss images. As you can see, most of the plasmids, they're not a nucleus. Right? There's some very bright, that's good, but a lot of them are not. And that's why she lost her protein. Yeah? And well, now I have to use another imaging feature, which Joe mentioned as well, is the diffusivity, if I say it right. And so it measures the total intensity versus maximum intensity. So, right, if you have the full size of those fluorescence and the diffusivity is low, right? If they're evenly distributed, then you have a high diffusivity. Okay, so let's see what they look like. Now, the plots, so now on x-axis, we plot the diffusivity, and on the y-axis, we plotted the radial moment. Okay, and then you can separate the two populations now. The ones with high diffusivity with the images, and you can see here, it's not about the intensity of GFP, right? So you do have very bright GFP that's, again, evenly distributed in the cell, which is good. But a lot of per assay, right, generated those GFPs. So not that many copies in the nucleus, but they are in, right? At least they're not on the two ends of the cell. So those are the ones actually with her protein in it, okay? And then if we look at the other population, those are the ones not good. Okay, so then we sorted, she actually asked, she wanted to verify, of course, that's good. So we sorted this population and this population. I haven't heard complaints yet, so that's good. So it's good, she got her protein. And okay, so that's one example that we're really happy with the one assay that we're very happy with. And the other one is another very difficult sort, right? Labor intense too, that's neuron. I don't know how many of you work with the neurons. They are very difficult to work with, they're very fragile. Right? We have been sorting these neurons on ARIA for a long time, use either 100 micron nodes or 130 micron. They're okay. But however, just because of the assay, right, you always get a lot of debris. And in this example, right, we use live and dead, and these neurons are retrogradely labeled with the wheat germ agglutinin, right, labeled tagged with 488. So these, for 2 days, right, they injected his eyes for 2 days, the dye got uptake and by the neurons, and they will the neuron body would have the protein take up this protein and be green. So we sorted this population. It's very low. You see that under 1%. And when they pick up the sorted cells, which we think that those should be all positive neurons, right? And what they told us is that, unfortunately, it's still a mixture. It's a mixture of neurons and also a lot of debris that took up the GFP. Same thing. Yeah. So what happens afterwards is that their technician, she was really funny, and she was like, yeah, after you finish sorting, then I'm pretty much married to my microscope. Like she would take 6 to 7 hours sitting in front of the microscope single-handedly and pick up this, single neurons and deposit into those 96 well plate and lyse the cells and send for RNA sequencing. It's horrible. I said, I do not ever want to do it. So then, so we actually, sorted these cells on assay. So here's some images. As you can see, very dirty, right? Yeah, you've got all kinds of stuff in the assay. Some clumps, some axon, dendrites, and there's some nice round neurons. And but the majority of our debris and something else. So now adding the GFP, let's look at the GFP, right? So we did, again, using the same same methods, right? Like I just show you the diffusivity and radial moment, and we can identify the cells that actually have the big cells with the GFP uptake in their neurons. So now we can sort this one, and actually discard the other ones. and they can just collect everything in the plate and send over to the RNA-seq and seq core. And we can also, because the assay also has the index sorting feature, we can come back and match the cells that's been deposited and sorted on the plate. So, so the PI basically told me, we're only gonna sort on assay from now on, not even ARIA. Yeah. So anyway, so that's pretty much all the samples that what I want to show you. So I hope that by looking at these images that I can give you an idea how powerful these images are. Right? It's really, that's showing us something we can never see before and never do before. So here's a quick summary, right? Only S8, again, it's 6 imaging detectors, only on 488. So you have to keep that in mind. It's not an image stream. And there are many other, many very useful image-derived parameters that we can use to identify specific cells. And I only took out the ones that that's mostly useful to us, and there are many, many more in the system. So if you're interested, you can go talk to people at BD booth. And then for the sorting part, the other thing that's sort of kind of my users are a little bit reluctant at the beginning to use assay is because they think that, oh, because now you're adding these imaging features, the sorting is going to be very slow. And no one's, of course, we charge in their mind, right? If I can get the sorting done in 30 minutes, I'm not going to spend extra $200 for the time, just to get the images, even though the benefits are there. They still don't. But the good thing is on assay, once I show them how fast it is, and it's a no-brainer. So on assay, you can still sort very fast, almost as fast as ARIA. I'm not talking about lymphocytes. Lymphocytes, you can just push it through like crazy. Yeah. And so last but not the least, and I'd like to thank everybody at UT Health San Antonio Flow Core, Mike Burton and Sebastian Montagnino, who are here at the conference. And Kathy Davis, unfortunately, she could not be here in person. So without them, I wouldn't be standing here. And also, I'd like to thank our the VPR's office for all their support, as well as Mace Cancer Center. And also, we have funding from NIH and CPRIT. And the last thing, I hope it's okay that I want to mention is that we're expanding. And and we're hiring. So if you're interested or you want to move to San Antonio, please feel free to contact me. Thank you. Oh, okay. Yeah. We're running. Oh, it's okay. So how do I? It's on.
Thank you, Yulai. And Joel has asked me to mention that he's hiring too. If you want to move to Houston. Just kidding. Okay, so our last speaker for the day, I think fits really nicely into this, and it's kind of great to see how we're able to take these new technologies that are introduced and then apply them to some really cool research. And so Ken Hu is our last speaker, and he's done exactly that, except he invented one of these technologies, which is called ZipSeq. And we'll see it's another single cell technology. I'm glad that Bartek brought that up, that cytometry really has to do with all kinds of cellular technologies and not just flow cytometry explicitly. But anyway, so Dr. Hu is a fresh assistant professor at MD Anderson Cancer Center, and he's also an assistant member of the James P. Allison Institute, which is newly founded, and he's a CPRIT scholar. He's did his PhD at Stanford with Dr. Manish Butte, using atomic force microscopy to probe force generation by live cells. And then he did his postdoc at UCSF with Dr. Matt Crummell, where he invented this technique, ZipSeq, for applying DNA barcodes or zip codes to define regions of tissue and allow them to provide spatial context to single RNA sequencing data. And he's going to show us some of that today. I've seen the talk before, and it's very cool. So I hope that you guys enjoy that. Okay.
Thank you. Thank you, Sarah, for the very kind introduction, and thank you to the event organizers for having me here. I'm happy to be here to talk about some of the work I did at my postdoc, and then kind of give a hint for where we're taking that now in my own lab. So a little background: I'm assistant professor, as Sarah said, in Department of Immunology at MD Anderson. I started about May last year, so it's been a while. It's taken a while to get the lab set up, but we're up and running. Excited to get started. Right. So to motivate things, and I've prefaced kind of a disclaimer here, nothing I show here actually, I'm not showing any flow data here today, but it is all single cell data, nonetheless. So to kind of motivate the kind of thrust of my research and my vision is, if we consider a tissue as a multicellular structure, it has kind of 3 dimensions, and then if you consider time as the 4th dimension. And if you're a cell in that space, it's particularly immune cell, they tend to be highly plastic and migratory, which means they can actually see a diverse set of environments. So imagine this tumor or tissue, tumor is a type of tissue, as a heterogeneous structure. And if you're given an immune cell in that 3D space, you could see a very different environment. And these kind of environmental cues could come from things like the extracellular matrix, your proximity to certain things like blood vessels, cytokine and metabolite gradients, and then proximity to other cell types that you might interact with. Now, we can get at these kind of 3D features pretty well using imaging technologies. And here you can see just a clear, thick piece of tissue, in this case, a tumor, looking at a couple features of interest, like I hinted, blood vessels, and then the gradients of certain metabolites coming from them, and then the localization of different cell types in here. You can appreciate, if you're a cell and you're exploring the space, you're going to see a very different environment based on whether or not you're here or here or here in this tissue space. And kind of on the flip side, if we want to quantify the state of a given cell population within a tissue, we now have very good tools for that as well. Namely, obviously through flow cytometry, but also now you can dissociate your tissue and do things like single cell RNA sequencing, and then kind of project that data into a low dimensional space. As that was mentioned before, very nice primer on how maps work. I have Tisney here, but same thing, same idea. But the problem here is that this approach necessarily means you dissociate your tissue. You've lost that spatial context for where this given cell, this cell was, this cell was, this cell was, this cell was. What we'd like to do is, first of all, be able to place those single cell transcriptomes into a particular location within the tissue, and then overlay from imaging those features that we imaged of interest, and understand how that kind of the cell's unique expression profile and is kind of the overlay of all these signals that it might be seeing in its local microenvironment. So we kind of have to tackle this from multiple fronts. One being that we have to be able to link that single cell expression profile to position in the tissue. So kind of, taking these, if you imagine each of these as a profile, each of these like as genes, and we have to localize these guys at different positions. Then we need to be able to also decompose that cell's very high dimensional kind of profile into the summation of a bunch of gene programs, in this case, collections of co-expressed genes. So as Sarah mentioned, our approach for kind of the objective number 1 was to develop this technology called ZipSeq. And the idea behind this is, if you're familiar with any single cell RNA sequencing now, one strategy you can use to reduce your costs, and kind of avoid batch effects, is to hashtag your sequences. So what if you could attach these hashtags in a spatially determined manner? Then you can also hash your cells based on their location within the tissue before you dissociated it. And so the magic happens here with this kind of DNA barcode that's attached to some kind of anchoring moiety. In this case, for a lot of the stuff you're going to see here, it's an anti-CD45 antibodies because we're interested in the immune compartment of the tumor and other tissues. And the magic really happens here with these NPOM caged overhangs right here. And what this signifies that the thymidine within this overhang is covalently linked to this NPOM caging group. And now normally DNA, if it's reverse complementary, it can hybridize. And that's what we did with these zip code sequences. So these guys have the reverse complement of that overhang and then kind of a handle that can be read out by the single cell RNA sequencing platform. Now, normally this O1 prime and O1 can anneal, but with the presence of these caging groups, that prevents that from happening. But then if you were to expose it to the right frequencies of light, then those caging groups will fall off and you get that hybridization event. Then the hardware aspect of this is you just need some kind of microscope that allows for spatially modulated light illumination. So you can image, determine your regions of interest, zap them, and then anneal the barcodes. And here's just a really quick proof of principle. You have a tissue section, in this case a lymph node, which is just chock full of immune cells for people who aren't familiar. And what we can do is we can sequentially print on two regions of barcodes, one green region, one red region. In this case, green signifies kind of barcode, red signifies barcode 1, green signifies barcode 2. These are just these DNA molecules with a fluorophore attached. This is just so you can visualize it. But obviously, sequence space is much larger than fluorescence space, fluorophore space. So what we'll be able to do, oops, a little bit out of order. This is a quick demonstration of how it might work. You can imagine if you have immune cells kind of embedded within your tissue. Let's say I want to be able to distinguish guys from this region versus this region. There's obviously no physical features here. It's just kind of arbitrary at this point. But let's say we're interested in this region. You can select it and zap it. Photocaging groups fall off, and the barcode anneals, and then you can wash it off and repeat it. And this way, in end cycles, you can find cells with tags from end regions. And then on the back end, when you pass these cells through your single cell encapsulator, you can get a single cell transcriptome, as many people have done before. But you also, in parallel, you also get a zip code library that tells you, oh, this guy mapped to this location, and this guy mapped to this region. So now you can localize a given single cell transcriptome to a region that you can find. In this instance, we're going to look at the lymph node. So the lymph node really is a hub for a bunch of immune cell populations to gather together. There's a lot of heterogeneity in the cells you see there and the states that they can occupy, but the structure of the lymph node is also intimately tied to its function. And you can see here in this example, we see two lymph nodes. On the left is an untreated lymph node just at rest, kind of baseline, and then on the right is one that's been treated, kind of the draining lymph node from LPS immunization. So you get basically it's a very large inflammatory signal. And then you can see here we've also, this is a whole cleared lymph node. So we're actually seeing this 3D space. You're seeing these different immune cell populations, and you can appreciate that they have their own particular localizations. One of the most notable things is that upon the inflammatory signal, a lot of these populations kind of get pulled towards the center of that lymph node, the core of that lymph node. Now, imaging is great and all, but we're limited in the channels that we get. We can tell that these guys are CD4s or CD8 T cells, XCR1 positive dendritic cells, B cells. We don't actually know within that population, but how does their gene expression diverge based on where they're located in the tissue. So we kind of took these sections out of that, and we were interested in this. This is one of those lymph nodes post-immunization. You can see here that dense core of the lymph node that I talked about, where we pulled in all those XCR1 positive dendritic cells, and also these T cells marked in red. And then kind of this we defined so we defined that as region 1, or we're going to call core. And then there's the outer T cell zone in green. This area actually looks a little invisible because we have no visualizable markers for that. But nevertheless, there's many cells embedded in there. And then kind of right outside, we also have CD19, which marks kind of the B cell zone of the lymph node. So with these kind of 3 regions that we defined, this was a nice proof of principle experiment. We can look in the transcriptional space. Here's a UMAP, which you've seen before today, where you can kind of place your different cell populations into different clusters. You have your B cells, plasma cells, your dendritic cells, broadly speaking, certain myeloid populations, and then a lot of then your T cell mass over here, and plus this little proliferative T cell island. Now, what you can do is now overlay that regional kind of localization on top of that, and appreciate that there's these different clusters have very different enrichments towards different zones that we've defined. Obviously, B cells are largely put in the B cell zone. It's not too surprising. T cells are largely within the T cell zone, which is red and green. And then if you look at these dendritic cell populations, they have kind of different enrichments as well. And then this proliferative T cell population is very, very much biased towards the red, that core region. So we can, on a broad level, say, these different clusters localized to different regions with different enrichments. But, how do other cell types partition with them? And what if we zoom into a given cell population, look at it in a little more detail? So let's look at dendritic cell subset. So what we want to do is, in this continuum of dendritic cell states, we want to kind of pull out gene programs, which is collections of genes that are co-expressed. And we can do this because we have all those thousands of dimensions that we could obtain from single-cell RNA sequencing. And this is what the regional localization looks like on top of that. And if we look here and kind of pull out these gene expression profiles, we can find a couple of these programs, and these kind of blocks of genes you can see here represent these programs that we're kind of pulling out in an unbiased fashion of genes that tend to go together by expression level. And we can see one program is called we're going to term CDC1 program. Includes things like IRF8, which is a transcription factor that drives them into the state, the CDC1 lineage, and they're largely found towards the center, the core regions of lymph nodes. This matches, what many people have seen, which is that on this immunization, CDC1s tend to go towards that center core area of the lymph node, and that's where they also prime. They're able to execute one of their main functions, which is to prime CD8 T cells, which are also in that region, and you'll see that in a moment here. And then meanwhile, CDC2s kind of they often present to CD4s, and canonically they kind of sit just peripheral. They're still within the T cell zone, but just outside that core region, and we see that enrichment in that green zone. And then you can see these other programs with their own kind of unique localization profiles. And now within the T cell population, we can pull out, interestingly, that if you see this island right here, it's very much enriched towards that core region. And if we look at the cells that we transferred, which were those red cells, we actually that red marker was a fluorophore, which was being expressed. So we can actually pull that out in our transcriptional profile. Every cell that's marked here in red represents one of them. And those T cells are unique because they actually are specific to the antigen that we're vaccinating against in that experiment. So that LPS injection for the immunization was also co-injected with ovalbumin, which is chicken egg ovalbumin as a model antigen. Those T cells marked in red here are specific for an epitope within ovalbumin. And you see they're largely localized to that red region here, which matches what we saw with our imaging. So, it's a very nice kind of correlation of the ground truth that we obtain through imaging and the data that we get out using this ZipSeq approach. And when we do this gene program analysis, what we find is that programs involved in biosynthesis and replication are very much enriched towards the T cell core, which makes sense. The T cells there, CD8 T cells are being activated. They need to start proliferating and undergo biosynthesis. And then kind of as we move towards the more periphery, we see them adopt more of a effector state, and also CD4 Treg cells kind of are localized just outside of that, a little more outside that core zone. As we move towards the outer regions of lymph node, in the B cell zone, we see more of a Th17-like gene program be adopted. So this was a nice, proof of principle experiment to show that, in a model system where we know a lot about the compartmentalization of the cells and how that ties together to the function, we could actually faithfully recapitulate that using our approach, and combine, and shows the strength of combining imaging with single cell transcriptomics. And then in the tumor setting, we were interested in, this is a much less organized tissue. We were interested specifically in this melanoma model. And so in this model, the melanoma cells themselves are going to be marked in red with M cherry. We inject them into this mouse model where we're specifically interested in a subset of macrophages that express CD206 or MRC1, which marks a kind of an M2 suppressive state. And what you can see here is that when we inject these tumors and then harvest them and section them, the tumor itself, there's masses marked in red. And then you can see in green kind of these macrophages, these CD206 expressing macrophages. And you immediately notice that there's a rim kind of population that's right on the edge of the tumor, just outside, very, very high in reporter expression, and kind of a more diffuse localization of these cells towards the interior of the tumor. So looking at that ratio of red to green, we kind of define three regions, inner, kind of an outer, mid, and inner, or red, green, and blue. And we were able to pull out our immune cell, single cell transcriptional profiles, and immediately appreciate that certain populations have different enrichments towards the center of the tumor, towards the edges of the tumor. Within the CD8 T cell population, we see this kind of a TCF7 positive, this population that's located more towards the exterior of the tumor, and then ones that start to express some of the beginning markers of exhaustion located more towards the interior of the tumor. We're going to focus on these retinal positive macrophages. So this island right here of cells is very much enriched towards the exterior of the tumor in this very thin band here. And so one of the genes that differentiate that population is retinal or realm A. And we stain for that. And indeed, really, if you look at CD11B myeloid cells, really the only ones at the edge actually express realm A. And it's really quite spatially restricted to that edge. And you can see these more myeloid cell populations towards interior, they lack that expression. So it's nice to see by independent, imaging approach that what the data we're getting out actually is validated. Now we are interested in exactly what these retinal positive macrophages are and what their implication is for human health. Problem is, retinal is actually has no human ortholog. So you're like, okay, what relevance does it have for humans? Well, in literature, previous papers have shown that this retinal population of macrophages also expresses other genes, and we can get at them because we have their whole entire transcriptional profile, or at least the genes that didn't drop out. And in several papers, they identified that macrophages with this very similar profile, which is retinal, and also FGFR1, and they're high in MRC1 and high in FOLR2, but low in CX3CR1 expression, as CADM1 expression. They found these particular macrophages associated with kind of aging and fibrosis. And then in another paper from Julie Helf's group, they found that in human and mouse breast cancer models, that this particular macrophage population with this unique expression profile was associated with better clinical outcomes in humans and greater CD8 T cell infiltration. So really, these seem to be actually good, potentially good guys in terms of promoting the anti-tumor response, which stands a little bit in opposition to traditionally what people have viewed is that MRC1 or CD206 represents a marker of these suppressive M2-like macrophages, which you wouldn't want in an immune response against cancer. What they also found was that these guys are more stromally localized relative to TREM2 macrophages found in the interior. So this again matches what we found, which is more stromally localized, meaning just outside of the tumor mass. That matches our localization data from ZipSeq as well. So this work was then in collaboration with Rajwa Ray, back in my postdoc lab at UCSF. He created a reporter mouse the construct I showed you earlier. It also includes a DTR, which allows us to specifically deplete these MRC1 expressing macrophages and see the effects of that on the overall immune systemic response. And when we do that, when he did that, first of all, those guys are gone. You can see these bright green guys at the edges of the tumor. They're completely gone in the DTX treated model. And if you look at via single cell RNA sequencing, those retinal positive macrophages have been completely eliminated in the plus DTX condition. Now, this actually is flow data. Rajwa was able to look at the frequencies of different immune cell populations, traditional flow, and found that, when you got rid of these supposedly immunosuppressive macrophages, you actually got greater CD8 T cell infiltration of that are specific against the tumor, these OT1 CD8 T cells, which stands in opposition. But it actually matches what we saw before, what other groups have seen before, that these unique retinal positive edge macrophages actually promote CD8 T cell infiltration. So when you get rid of them, we get less of them, as of less frequency of them. And indeed, also when we removed them, we actually found that the T cells that did remain actually expressed higher levels of these markers of expression. Now, this kind of leads into the next part, which is, where are these macrophages coming from and who are they interacting with? Because if we can figure that out, potentially we have a lever with which to promote their presence and then promote kind of downstream CD8 T cell infiltration to the tumor and better responses, immune responses against the tumor. We got interested in fibroblasts and their interaction with macrophages through some wound healing studies. So, we're like, can we capture my fibroblasts from the tumor? Problem is, fibroblasts aren't immune cells, so CD45 antibody won't target them. So we kind of had to adapt our ZipSeq to an anti-MHC class 1 antibody for ZipSeq. And doing that, we were able to capture these fibroblasts as well, and found that their gene expression varied as well based on their localization within the tumor. You get, for example, you get more IL-34 expression towards the interior of the tumor, and then on the edges of the tumors, these fibroblasts have expressed things like podoplanin, LOX, and CXCL12. We used a program called CellChatDB to basically look at ligand receptor co-expression to try to predict cell-cell interactions. And normally what happens is you have a mix of cells. If one cell happens to express a lot of the ligand and another one happens to express a lot of the receptor, you might predict they're going to interact. However, you don't really know where they were. But now we have the ability to say, well, they're within the same kind of anatomical region within that tissue. So you can be a little more confident that they're potentially interacting. When we did this, we found a very large enrichment, actually, of signaling axes between these retinal positive macrophages and the fibroblasts that exist also at the edge of the tumor. And you can see these signaling pathways have different strengths based on within which region we're looking at. IGF signaling is predicted to kind of stem from the retinal maxis senders and the fibroblasts as receivers. And then kind of reciprocally, we found that FGF signals were predicted to be generated from the fibroblasts and they're received by the retinal positive macrophages, possibly through FGFR1. And indeed, the edge localized fibroblasts preferentially express these FGF family members. We were also able to show, oncostatin M was another signaling pathway between monocytes at the edges of the tumors and fibroblasts. And all of this to say that our final model that we constructed that we're testing now in the lab, that was all enabled by the spatial dissection of the tissue, was that at the edge or the stromal kind of neighborhood of the tumor, there exists this kind of interaction between monocytes producing oncostatin M, activating fibroblasts to produce FGF family members, which can promote the presence of these retinal positive MRC1 high macrophages in this kind of feedback loop potentially. And then you get this kind of, then through unknown mechanisms, they seem to recruit and promote the greater frequency of CD8 positive T cells. And kind of towards the edge, interior of the tumor, we have this other not so good interaction potential between exhausted CD8 T cells, fibroblasts, and these TREM2 CX3CR1 high macrophages. And this was from another publication that I didn't want to get into today. But kind of our goals now are to come in and start genetic perturbations to eliminate these different populations and try to stimulate the fibroblasts and actually genetically perturb them in situ using delivery of guides in CRISPR models. So in summary for this section, ZipSeq allowed us to merge single-cell RNA sequencing data to imaging data. And with that, we can describe differential localization of broad immune cell populations, within a tissue, but then also within a given population, like T cells, macrophages, these broad populations. We can actually see how gene expression varies based on where they were. And then that spatial context gives us greater confidence when we're trying to predict cell-cell interactions, when we infer through, pure inference based on transcriptomics. are we okay to go over? Okay. In that case, I can get a little bit at this part 2, which is again, getting at this idea of applying spatial and temporal context to single cell RNA sequencing data. We kind of made a little divergence from tumor world to wound healing world. And you may ask, why wound healing, study cancer. Well, it turns out there's a lot of similarities between the process of wound healing and tissue repair and tumor growth and progression. Indeed, there's a famous quote that tumors are basically the wounds that do not heal. So we really want to put that to the test. And you can imagine in kind of a phase space, your wound healing causes a insult to the tissue and it returns to a new homeostasis. And whereas tumor growth might resemble that in some ways and then diverge. So to get at that spatial and temporal those dimensions of the wound, we created these full thickness wound punches within the back skin of mice. And what happens is wound over time closes up and heals. But there's a natural geometric, there's a natural kind of geometry to this wound in that you can sample different radii around this wound. You can sample using this kind of radial hole punching method. We could sample like just the center and just outside these rings. So we sampled 4 of these rings and we looked at them 4 time points post wound healing. We looked at both the immune and non-immune population, and we could barcode them and then for multiplexed single cell RNA sequencing. And we got, a lot of the main immune cell population that you might expect, a lot of myeloid cells, a lot of neutrophils, some lymphoid cells, dendritic cells, and whatnot. And you can also see how they evolve, the distributions change over time. So day 0 means the unwounded state. So this mouse it's just a normal intact skin. That's what the distribution looks like. I'm sorry, and we're going to zoom in on the macrophages. You can see the distribution down there is what you see at unwounded, and then day 1 immediately you see this massive shift towards the left side of this UMAP, and then as you progress through time, they kind of move through that curve, and then day 14 you start to return to what seems to be more of a normal distribution compared to the day 0, but no, quite the same. So to describe this kind of data in both dimensions of space and time, we need a new way to look at things. So we kind of create these space-time tile plots in which we have space on the y-axis. So to the center punch, then just outside, and then just outside, and then just outside. 2, 4, 6, 8 millimeters. And then the days post-wounding. So day 1, day 3, 7, 14. And then, of course, the unwounded state is represented in the color. So whatever parameter you're looking at, it could be like the frequency of a certain cell population. It's basically the color represents its value pinned to the unwounded state. So if it's bright red, that means it's much higher than the unwounded state. Blue means it's lower. And this is, I think, an intuitive way to try to look at these space-time profiles. And now you can look at these different immune cell populations and their subsets and look at, they have their own unique profiles. Like this guy is enriched right at the beginning of the wound, right just outside of the edge, the wound area. This guy's enriched right after wounding, right in the middle, and so on and so forth. This one at day 3. This one actually starts to pop up just outside the wound at late time points, and so on and so forth. And kind of on a holistic level, we did a bunch of dimensional reductions to try to look at the overall, flavor of all the immune cells there. We can see this very nice arc. See here, as the wound progresses and heals, it's trying to return to the unwounded state, but it actually seems to overshoot a bit. And if we would like to take this out maybe a little longer to see if it ever does return to something resembling the unwounded state, or if it kind of is ever so slightly shifted off, and that you don't get complete restoration. And then, of course, the non-immune compartment has its own particular cell types. We were interested in fibroblasts. And I'm going to rush through this part relatively fast, but this is to say, we're able to verify some of these gene expression space-time profiles through imaging. We are able to take big pieces of the wound and clear them and stain them using a variety of antibodies and also endogenous fluorescent reporters. And you can see things like this periostin gene expression. It really spikes during day 7 in the center of the wound, and that's indeed what we see. You can see here, this is kind of the radial dimension moving out from the center of the wound. See, there's a lot of expression at the center, and it drops off pretty fast. So that matches our space profile here. Now, as I mentioned, we want to be able to pull out computationally gene expression gene programs. So we use non-negative matrix factorization, which is a way to get at describe high dimensional data sets. So if you have a big old matrix of cells by genes, you can decompose that into the product of two smaller matrices, where it's one matrix is cells by what we call factors or programs. The other one is programs by genes. So this represents kind of the level of each program within each cell. This matrix represents kind of which genes contribute the most to that given program. And if you look at these programs and their levels over space-time, they also have their own particular profiles. And then if you look at macrophages, let's say in the macrophage, you can see they have their own, they have their own profiles as well. Like this one is right at the center and early on. This one's late in the center. This one actually kind of is just outside and seems to move into the tumor. Oh, sorry, not tumor, wound as it heals. So they have their own unique profiles. And you can, dissect out which genes contribute the most to a given factor. Now, what we're interested in is you may have noticed that some of these tiles might look a little similar in their between different cell populations. So we hypothesize that maybe that represents an interaction. These programs are linked. Maybe the product of one gene of one program in cell A is actually turning on program B in a program in cell type B. So there's kind of these interlinked programs that exist between cell populations. And when we looked at fibroblasts and macrophages or monomacs, we found 3 of these distinct pairings that we followed up on. And just make a long story short, we verified them. One that was oncostatin M from fibroblasts, from monocytes to fibroblasts in the early wound. And then there's 2 that come from fibroblasts to the macrophages, to nascent C, periostin. And using our gene program analysis, we could predict which genes will be turned on. And we verified that via several in vitro tests. We'll skip through this, but all this to say that this allowed us to basically predict cell-cell interactions that exist between different cell types in this very, very complex system with many, many cell types, kind of changing and working together over space and time. And then we had a bunch of other cell types too that we never really followed up on in our publication, but you can look at what these kind of blocks of correlation, and many of these programs span multiple cell types. So there's several keratinocyte factors and fibroblast factors, dendritic cell factor, endothelial cell factor, all together, these blocks. And this represents kind of an atlas of potential interactions and interlinked gene programs between different cell types. And then kind of the final part of this was our goal, theoretically, if these gene programs represent a fundamental unit of gene regulation, it doesn't matter what setting the cell is in, if the macrophage is in a wound or in a tumor. Those same sets of genes should be co-regulated and co-expressed and go together. Like one gene is up, the other gene should probably be up, or one gene is down, all other genes probably going to be down. So we decided to use this gene program as a tool for cross-context analysis. And this is illustrate that you can pull out these blocks, and then in the tumor setting, you can pull out the same block of genes, and maybe in one setting, both of them are high, and in another setting, this program is absent in macrophages and present in the wound healing setting. So we went to a tumor world, looking monocyte macrophages there, extracted gene programs, and then what we're able to do is by looking at the gene weights, the genes that contribute the most to a given factor, in a totally unbiased fashion, look to see which ones had the most overlap. And if there's no overlap, they'll kind of look like this. And when we looked at them, we found a couple that seemed to line up pretty well. And when we looked, and if you look at that scatter plot, here's one for interferon signaling. This is one of the strongest. It makes a lot of sense. It's a very well-studied pathway. A lot of genes that are very strongly turned on by interferon signaling, and so therefore you see they all line up very well here. It's kind of correlation. And then there's some that have kind of a slightly weaker correlation, but nevertheless were of interest to us. Kind of these EDGE MHC class 2, this antigen processing and presentation program, and then this kind of triglyceride sequestration. There's a little bit of noise, and I think this kind of boils down to the fact that we're limited in the kind of sampling of genes, and due to dropout of transcripts from our different single cell runs. And then this represents a situation where we absolutely had no overlap. Here's some others that we identified. These are all within macrophages. And basically what this allows us to do is make several predictions about the tumor microenvironment. We found was that factor 6 was very much enriched in macrophages from the B16F10 melanoma model versus MC38, which is a CRC model, in mice. And when we looked in the wound healing, this corresponded to wound healing macrophage factor 22. And one of the things we found was that that was associated with fibroblast factor 10 or gene program 10. And that was mediated by periostin. So we hypothesized there's more periostin signaling to macrophages, the B16F10 setting than MC38. And that indeed seemed to be the case based on our immunofluorescence imaging. You see more of these periostin kind of fibers that are being secreted, being generated by the fibroblasts, and they tend to contact, your myeloid cell population more frequently. And we can also see that in 3D as well, where these kind of these dense like networks of periostin are being deposited. And we found some of these programs can be conserved across species and humans as well. We found this same pairing conserved in human samples, and they also are correlated in their levels across head and neck squamous cell and lung cancer models, limited data. And then in the time aspect, actually, we also found that as the tumor progresses, we looked at a couple publicly available data sets of tumor progression. We found that as the tumors progressed, and if you look at the gene expression profile, the fibroblasts within them, they actually start to look, if you embed that into the wound healing world, and describe in terms of wound healing coordinates, as the tumors progress, both this KPP, this pancreatic cancer model, and this, again, B16F10 melanoma model, they all progress towards the day 7, 4, and 2 millimeter coordinates, which is to say that as the tumors progress, the fibroblasts start looking more and more like the fibroblasts you would find right when the wound is closing up and it's starting to kind of resolve the inflammation and remodel the extracellular matrix. And that was driven by some of these factors like programs 8 and 10 within the fibroblast. So in summary, cell types such as macrophage and fibroblasts, they really occupy a diverse continuum of states. And when we try to cluster them, it's not really doing justice to the diversity, to the heterogeneity of these populations. So we can decompose them as the summation of many, many gene programs. And then we can examine the space-time profiles of these within a highly dynamic system such as healing wound and predict cell-cell interactions. And this also allows us to look for conservation across different tissue settings from tumor and wound. So just acknowledgements. I'd like to acknowledge my, of course, Max Krummel's group where I did most of this work as a postdoc, and many of my collaborators here. There, where our imaging facility, a core facility there, also our flow core facility over there, which they obviously very helpful for a lot of the work that we did, and hoping to, of course, make use of the flow facilities around here. Now that I'm around, and then of course my own lab, which currently consists of 2 staff members, Sarah and Jehu, and then we're always on the lookout for talented students to the GSBS program and potential postdocs, and the core facilities at MD Anderson, which we've begun to make use of, and of course our funding sources. So thank you.
If people have specific questions, they can ask, but we also have a roundtable scheduled. So if you want to hop in for that, you guys can have a seat and then we can incorporate all kinds of questions.
That was a really great talk. Thank you. I was just curious, and you may have mentioned this at the very beginning and I missed it. For the zip scope, what is the resolution that you're going through those tissue sections to remove the blockage so that you can do that annealing? So what's the resolution of that labeling process?
Sure. Should I repeat the question for online audience or? Oh, okay. No, that's a fantastic question. Yeah, I didn't quite mention that. So there's a theoretical space resolution, then there's a practical space resolution. The theoretical resolution is determined by basically whatever modality you're using for spatial light modulation. In our case, we use a DMD, a digital micromirror device, depending on your magnification. You know, like the 20x magnification, you can select a single cell. However, functionally, functionally, you have to be able to recover that single cell during your single cell RNA-seq run, which is, you know, a dicey prospect based on, you know, losses during the whole process of sample prep, you know, digestion, and then encapsulation. So functionally, you know, we're not at the single cell level. We tend to go for these larger areas, as you saw, about 100 to 200 microns is kind of these microanatomical features. And again, you know, the purpose of this approach was not to kind of be able to, the purpose of the approach was to actually map single cells, not to a given XYZ coordinate, but to a certain tissue feature that you can image. So it really is the marriage of imaging and single cell RNA sequencing. But the regions that we define are kind of on that order. The kind of smallest regions we've done are like germinal centers within lymph nodes. So, you know, the widest dimension, we're about 170 to 100 microns, I'd say, in size. Yeah. Thank you.
I'm wondering, given where you were working, if you guys also, when you did the macrophage depletion, if you looked at response, the impact of tumor growth, and then also tumor response to any anti-tumor therapies?
Yeah. What generally we found, I should specify Arjo found, and again, if you really want to follow up on it, he has a bioRxiv paper. I think I linked it, but if you just search Arjo, Ray, and Max Crummel and my name on bioRxiv, I'm sure you'll find it. Yeah, he found a very interesting phenomenon, which is that depletion actually did not. Normally, we'd think that, you know, it'd be helpful for the tumor. It would, you know, promote the immune response and prevent kind of slow tumor growth, but actually made the tumors less responsive to checkpoint blockade. Again, because we think what happens is you're getting rid of. So I should specify that the MRC1 reporter, those retinal positive macrophages edges are not the only CD206 expressing macrophages. There's many more towards interior that were also CX3CR1 positive and also TREM2 positive. And from what we know, those guys may not be so great. But I think what's happening is with that reporter, I mean, with that mouse model system, you're depleting everybody. So you're getting rid of good and potentially bad. So I think that's part of the, you know, the surprising thing that we saw. Yeah.
Same testing. This is a question about the S8. Have you guys had any thoughts about if it would be possible to measure 6 sort-induced cell stress with the imaging capabilities seen? Would we try different sheath pressures, stress the cells? Is something we can measure?
Oh, that's a good question. That's a real, so if looking into 6, so in the cell, we haven't really looked into the viability post. We did a little bit of work into, and we're going to look at the data. Obviously, we can't capture anything else, but into imaging and apoptotic, where if we can see it feathered. But once again, it's not quite as resolved as the amnus. So you're looking at it, you're right now you're doing kind of a variation off of that absorption, the scatter mask. That's not this is dark field, I think of, but no, it's it's the light loss. Oh, I always forget light loss.
That's the one.
So the light loss will give you that image, and it acts kind of like a bright field. Our one issue of analyzing it though is going back, doing the post analysis. We don't know necessarily what the sort decision was yet. So we've been kind of pushing them a little bit to create some kind of a mask so we can then see what the sort decision was and then go back later for better analysis. Yeah, and so right now our hands are a little bit tied because we can't go back retroactively and look at some of the data, but it should be in the future.
I think you pretty much summarized very well. And one thing I'd like to add is that in the software, if you're thinking about like you can change the pressure of the sheath, right? And it's all unfortunately already preset with the nozzle. So I'm not sure the software will let you do that. Yeah. Maybe someone from PD can correct me, but I think the only thing actually, my a little bit complaining about essay is really just the software. It's more rigid than ARIA, definitely, than DIVA, yes. Mhm.
Some other questions on the S8. So you saw like those fragments in the light loss images. I'm wondering if you saw that there was a correlation there between a specific nozzle, because that looks like continuing like vibrations, like do the piezo amplitude, but like at least I see sometimes with another like jet and air systems.
So say that again. So are you saying the particles within the stream?
Like
Or
in some of the light loss images, it looked like they were like wavelengths, like banding right after that. I'm wondering if you've seen a correlation between like specific nozzle settings to where that appears more or less. Because like even in your presentation, like in some of them it was more pronounced, but then in like some of the other light loss images, it wasn't.
Yeah, we found that for cells, it was more pronounced in cells, right, as opposed to beads. So you see the kind of ghost or those little visual aspects to it. But as of nozzle, I don't know if it's specific to nozzle. We'll go back and look, take a peek. But yeah.
The other one was the singlet gating that, and for other folks that have these imaging cytometers, I'm seeing more and more that there are cells that we are gating out with our traditional doublet gates that are indeed singlet cells. And depending on like the immune population, it can be like 5% sometimes. I noticed like in the gating strategy that at least when you showed it was the traditional gating and then singlet gating based on morphological information. I wondered if you've ever tried doing the morphological gating first for singlets to again see those images that are just singlet cells that are being gated out traditionally by doublet gates.
I see what you mean. No, we have not tried it. Yeah, but that's a very good suggestion. Maybe we should look. Yeah, maybe we're excluding something we shouldn't. Yeah, but for everything we've done, I think if you're working with some rare cells, I think that's really important, right? You don't want to lose anything. But for the assays that we've been working with, they always bring me so much, and they never really cared. But that's a great suggestion. Yes.
So when we were sorting, we did, there is singlets outside of what you'd consider singlet gate. And we saw that. Where we were hoping to look a little bit more into it, but ran out of time. But we would go in. And so we actually, I believe we eliminated one of our singlet gates at least because of it, because we had utilized the other alternative gate where they could do where you're using where the eccentricity, the elongation of it, at least in more circular cells. So that really helped a ton on your doublet. So you didn't have to worry about it as much. No. But now that I've seen your talk and seen that, now I'm going to probably start going back and looking at some data, not only that but on the site pics as well, to see if that is indeed single cells, falling out in in higher areas versus height, or in greater widths versus height or area.
Thanks.
I have a question for Dr. Hu. About those gene programs that you see in the macrophages. There's ostensibly gene programs that are differing between the CD8 T cells that are making contact on the edge of the tumor versus what they are inside. You showed, I think, in the talk that you have exhausted T cells tend to be towards the center of the tumor, and then like prime T cells are sort of outside in the periphery. I'm wondering if you've looked at those gene programs or those gene modules and how they differ between those two segregated populations, and then whether or not there's, is there any like common correlation between the two? Like, is it just that, oh, they're just migrating and they're getting away from some sort of signaling gradient, or are they actually encountering some other signal that comes from, let's say, the tumor tissue versus those macrophages on the outside that causes the gene expression program to sort of flip into an exhausted state? Kind of a loaded question, sorry.
Oh, sure. Let me see if so basically you're curious as to in that particular model, that B78 model, why are the T cells found more towards the interior kind of expressing more exhaustion markers than the ones exterior?
Yes, and is it
Okay.
related to those macrophages
Oh.
that you have on the periphery? Like when they make contact, does some gene program like start up, and then as they migrate, does that dissipate, or is it because they come in contact with some, like the tumor tissue, for example, it causes it to flip?
Yeah, that's a fantastic question. I think based on what we're seeing, our current working hypothesis is that these guys, those macrophages at the edge, help draw in kind of new T cells into the environment, and they kind of represent that they're coming in at a kind of earlier in the differentiation state, before they've kind of hit the tumor, so they start to pick up less of those exhaustion markers. So that's why when I showed when we get rid of those macrophages, the overall T cell compartment starts to express more of those exhaustion markers. Not because necessarily making them more exhausted, but you're getting rid of the ability to bring in new guys. So overall, the population is going to shift to kind of the ones that have been hanging around longer. Now, whether or not the ones that hang out longer are also the ones deeper within the tumor, that we can't say for sure. That requires like tracing, kind of doing some kind of the time-based tracing, which, there are certain ways to do that now with antibody labeling, IV antibody labeling, things like that. But we can just speculate and just say the ones that are potentially deeper within the tumor core have been sitting around longer, have either been exposed to more chronic antigen or potentially immunosuppressive cytokine signals, or also hypoxia, which is another major driver of this kind of T cell exhaustion. So any number of those features can combine to cause that. So at this point, it's a little speculative, but we think that again, that what we're seeing is we're shutting off the influx of new T cells to the tumor, and indeed that arrival of new T cells from priming locations such as draining lymphoid tissues is one of the main mechanisms by which a lot of checkpoint blockades work. So I think that this is potentially a fun, very impactful way to maybe boost the efficacy and explain why maybe immunotherapies don't work in some patients.
Cool, thank you.
Okay, well, we're right about at 5 o'clock. If there's no more questions, then I think we'll wrap up and thank the speakers. And we have another fun day tomorrow starting around 8:30. So thank you guys.